The remains of a 52,000-year-old mammoth revealed by a three-dimensional the structure of his chromosome. In 2018, in the Siberian tundra was perfectly found preserved remains of a woolly mammoth from 52,000 years ago. From a piece of leather the animal was able to read its full genome and reconstructed the three-dimensional structure of its chromosomes, which will allow to know the functioning the genes in them. An international team of researchers read the full genome and reconstructed three-dimensional structures of the hairless mammoth chromosome before 52,000 of years. This was possible thanks to the perfect preservation of the animal's body. It seems that the remains of the mammoth have undergone a natural process of freeze-drying, similar to that used for food preservation, also called drying sublimation. The conditions of the Siberian tundra 52,000 years ago allowed removal of water from the frozen body by sublimation of ice, i.e. ice passed directly from the solid to the gaseous state, without the liquid state. The three-dimensional structure of chromosomes gives insight into how the mammoth genome was organized and which genes were active in the skin tissue, from which DNA was extracted. The results and the description of the research appeared in the journal Cell. Mammoths, Mammothus primigenius, freely they walked the earth during the last ice age. It is believed that they died out about 10,000 years ago, although some secluded populations, as per Wrangel Island, has survived longer. Population from Wrangel Island is estimated it existed 4,000 years ago. And because they lived in a cold environment, many their remains have been well preserved, though none compare to it relative to the analyzed find. The lyophilized remains of the mammoth females are from northeastern Siberia and was dug up in 2018. The specimen contained not only bone, but also skin tissue. The maintenance process allowed to keep the body in its great condition. There is even hair on almost the whole body. Main fragments of ancient DNA can survive for a long time, Marcella said. Sandoval Velasco from the University of Copenhagen, co-author of the study. But what we found here is a sample in which the three-dimensional arrangement of these fragments DNA has been preserved for decades, so that we reconstructed the structure of the entire chromosome, he adds. It's a new kind of fossil, and its scale distills its sheer DNA fragments because it has 1,000,000 times more sequences, he says. Erez Lieberman Aden of Baylor College of Medicine, co-author of the publication. To also for the first time that a karyotype of any kind has been established for an ancient sample, he adds. A karyotype is a full set of chromosomes that find its in the cell. Knowledge of 3D genome architecture provides many additional information beyond its sequence. Most ancient specimens of DNA it consists of very small, mixed fragments, because DNA molecules they begin to fall apart when the animal dies, but in this case, to surprise the researchers, the animal's chromosomes have been perfectly preserved. Armed with exceptionally well-preserved remains, the team scientists extracted DNA from a sample of skin taken from beyond the mummy's ear. Then thereafter DNA was analyzed using a method known as Hi-C that can identify DNA sections that probably interacted with each other while the animal was alive. In ordinary DNA sequencing, its spatial is lost structure. Imagine that you have a puzzle consisting of 3 billion elements, but you do not have the final image of the puzzle, on the basis of which you could work, says Marc Martirinon, a geneticist from the Spanish National Center for Generic Analysis. Hi-C connects parts of the genome that naturally they touch, giving an approximate image of the whole puzzle. Researchers then combined information from Hi-C analysis from DNA sequencing to identify interacting DNA sections and create an orderly map of the mammoth genome, using the genomes of modern elephants as the template. Comparisons with modern elephants, closest living relatives mammoths, not only showed similarities in the number and structure of chromosomes, but also differences in gene activity involved in hair growth and adaptation to the cold. Analysis showed that hare mammoths had 28. The chromosomes are the same as modern Asian and African elephants. Scholars they also found that gene expression associated with the development of hair follicles was altered in the mammoth skin compared to the skin of an elephant. The last common ancestor of all life has already appeared 4.2 billion years ago. According to new research, life on Earth has already flourished several hundred millions of years after the planet formed. Scientists have determined that the common ancestor for all living organisms was a microbe that lived 4.28 billion years ago. DNA of all living organisms today has many similarities, suggesting that all life comes from one common ancestor. It is called the LUCA, last universal common ancestor. In a new publication which it was published in the journal Nature Ecology and Evolution International, a team of researchers led by researchers from the University of Bristol has determined that the organism that gave rise to all life on Earth may have evolved just a few 100 million years after the formation of the planet, much earlier than previously thought. What's more, he was more sophisticated than he suggested the previous assessments. LUCA is a hypothetical common ancestor from which it comes all modern life, from single-celled organisms, such as bacteria, after the bluefins or giant sequoias. LUCA represents the beginning trees of life before it split into groups recognized today: bacteria, archaea, and eukaryotes. A team of researchers compared genomes of a different group 700 of modern microbes. Scientists looked for common features to identify which ones came first. Although the analysis does not reveal how life was created, it suggests that a complex cellular organism has evolved as early as 4.2 billion centuries ago, just 400 million years after the Earth formed. This microbe had quite a large genome encoding about 2,600 proteins. He fed gaseous, H2, and carbon dioxide and had a residual system immunity to fight viral invaders. Our study showed that LUCA was a complex organism, not much different from modern prokaryotes, but what really interesting, he had an early immune system, which shows that even 4.28 billion years ago, our ancestor took part in an arms race with viruses. Davide Pisani, CO author of the study, said, "Our work combines data and methods from many disciplines, revealing insights about early Earth and life that cannot be founded was to gain in one discipline. It also shows how fast on an early Earth has created an ecosystem. This suggests that life can flourish in biospheres similar to Earth and other parts of the universe." Philip Donoghue of the University of Bristol. Scientists have previously been looking for LUCA. In 2016, the researchers under the direction of William Martin of Heinrich Heine University Dusseldorf, they applied a similar approach of comparing known microbes' genomes and established that LUCA was probably an anaerobic and a thermophilic organism. They also proved that it drew energy from hydrogen gases. This combination suggested he could live near hydrothermal vents. In these studies it was not established when the LUCA evolved. The research was based on variable but known indicators, genetic mutations in microbe species, as well as the rate of gene transfers between them. This allows you to create a kind of molecular clock. By constructing family trees showing which organisms are likely to be they evolved from others and tracking changes in genes, scientists may be more or less estimate when two neighboring branches have spread and thus determine age. It's their common ancestor. Martin's team analysis took this approach, which consisted of tracking changes in single genes common to all descendants. The researchers focused on 5 sets of paralox proteins or DNA or RNA fragments of common origin that arose as a result of the duplication. Such duplicates have been found in many bacteria and archaea, as it suggests that the chapter was still at LUCA. Tracking or mutation occurs in both copies of these genes, or only in one, makes it easier to determine their time duplications in the same age of common ancestors. To learn a little more about LUCA, the researchers traced 57 gene markers in 350 bacteria and 350 species of archaea. Separately, they followed evolutionary patterns of individual genes and gene families in these bacteria and the archaeals. Comparing the evolutionary stories of individual genes with stories species, they could better determine which genes were duplicated, lost, or the levels of gene transfer passed. On this basis, they inferred certain features the LUCA. In addition to eating H2 and CO2, scientists also found a gene that could protect the body from ultraviolet radiation. What it suggests that the microbe may have lived in surface waters where it captured CO2 and H2 from the atmosphere, not at the bottom near the hydrothermal vents, as determined Martin's team in 2016. On the other hand, they also found traces of the enzyme, which it is common in thermophils, which means that LUCA may have been also developed in a deeper environment. Researchers also found elements of the CRISPR-Cas9 defense system, the same one that became the inspiration for the most common genome editor used. Luca had this early immune system as a way to avoid viruses, explained Edmund Moody, lead author of the paper. It is up to Phi in turn. It suggests that there was already a diverse ecosystem 4.2 billion years ago, microbes and pathogens. The existence of a cave on the silver globe has been confirmed. It could be a lunar base. Scientists have long thought under the surface there is a network of caves and lava corridors. In 2009 it appeared publication describing about 200 cavities and pits discovered on the surface the silver globe, but it was uncertain whether they were tied to the caves. It seems that now we have been able to confirm this. For at least 50 years, astronomers have been theorizing about caves on the Moon. In 2009 a publication describing deep end pits discovered on its surface. The scholars speculated at the time that probably some of them are caves and lava corridors, created in the times when our natural satellite was volcanically active. Now a new analysis of data and photographs taken in 2010 year by Lunar Reconnaissance Orbiter, LRO, satellite seems to confirm the existence of an lava corridor in the Sea of Peace, Mare Tranquillitatis. The publication on this subject appeared in the journal Nature Astronomy. Theories about caves have existed for over 50 years, but after for the first time, we proved their existence, explains Lorenzo Bruzzoni, the University of Trento in Italy. How did scientists manage to confirm this? In 2010, as part of the ongoing LRO mission, Miniature Radio Frequency, Mini-RF, instrument, he collected data that included the pit in Mare Tranquillitatis. Many years later we re-examined this data using complex processing techniques, science that we have recently developed. We discovered radar reflections from the hollows that can best be explained by the subsurface cave and its tunnel. This discovery provides first direct evidence of existence an accessible lava corridor under the surface of the Moon, emphasizes Bruzzoni. The 4 or perhaps more sinkhole that astronomers have studied, it is one of over 200 similar structures previously observed on the surface of the Moon. These structures seem to be the result of collapsed lava corridor sealing under the influence of gravity or other factors, such as the meteor impacts. The pit of Mare Tranquillitatis is the deepest of the famous, so he seemed like the best place to start the analysis. It is about 100 meters width and sloping bottom, which can go further below the surface and can be an ideal place to build a permanent base there. Its size was large enough to detect any internal features with resolution, what LRO radar can provide. Simulations based on data collected by LRO, the new processing techniques, which showed that from the bottom of the sinkhole a tunnel or more lava corridor departs from 30 to 80 meters in length and the width is about 45 meters. Researchers believe the bottom of the cave is enough flat to be able to use it. Although from the surface to the bottom of the cave it is over 100 meters, with low lunar gravity it may not be. It's a big obstacle. Thanks to data analysis, we were able to create a model part of the corridor, says Leonardo Cara from the University of Trento. The likely explanation of our observations is the empty lava corridor, he adds. Due to the low gravity on the Moon, corridors' lava can reach a width of up to several hundred meters. Some can lead to even deeper caves that would be ideally suited for a shelter for the astronauts. The cave in the corridor, whose existence has just been confirmed, researchers, it's a good place to establish a lunar base. Future astronauts will need protection against rapid temperature fluctuations that the Moon experiences if they have stay there for longer. Constantly exposed during the day to the rays, the solar Moon heats up to temperatures of 127 degrees C. During the wind at night the temperature can drop to -173 degrees C. In the research from 2022, scientists have determined that in partially shaded pits and caves temperatures are constantly maintained for human functioning the organism. Protection will also be useful from meteorites, because on Silver Globe has no atmosphere to provide such protection. It is also important the cosmic radiation. The Moon has no atmosphere or magnetic field. That is why the astronauts on its surface have to reckon with about 200 times with more cosmic radiation than humans on Earth. Man's ancestors came to Europe around 1.3 million years ago. When and by what way the first hominids came to Europe from Africa? Redistortion of the 5 fossils from Spain indicates that they are about 1.3 million years old, which is they are the oldest found in Europe. On the other hand, the place finding indicates that our ancestors reached the old continent by the Strait of Gibraltar. Remains of the hominids found in 1982 in Ors, Granada provinces in Spain are the oldest in Europe and have about 1.3 million years. They are older than other remains of hominids from Eastern or Central Europe, what supports the thesis that our ancestors came to Europe through the Strait Gibraltar. Result and description of the research carried out by scientists from University of Barcelona, Berkeley Geochronology Center and Murray State University has published itself in the journal Earth Science Reviews. Dating was based on analysis magnetic properties of rocks in which the bones were found. The rocks containing ferromagnetic minerals retain the record of the intensity and direction of the field magnetic, in which they formed. Comparing the obtained data with the former indications of the magnetic field, the age of the object can be determined. Scientists were able to identify 5 events magnetic, which allowed them to link 3 positions in Ors with presence the ancestors of man. The oldest turned out to be the position of Ventimicin, where traces of hominids are 1.32 million years old, followed by Barranco Leon, 1.28 million years, and Fuente Nueva 3 to 1.23 million years. Scientists also analyzed the remains in their research fauna found at various sites in Ors and compared them with those found in other early Pleistocene sites in other parts Europe. The results indicate that a small and large fauna from Ors is more primitive, for example, the one from the position of Cima del Elefante in the Itapirca mountain range in Spain, where evidence indicates that the rodent Alephiomis lavocati is more developed than Alephiomis found on positions in Ors. Another important indicator of age is the lack of remains pig ancestral ancestry. These animals are considered to be Asian immigrants and not they were found on any excavations in Europe from 1 to 1.5 million years back, while they were found at Cima del Elefante, which confirms that the fauna from the excavations in Ors is older, explains Luz Gibbard from University of Barcelona, lead author of the publication. These studies indicate that early hominids arrived in Europe through the Strait of Gibraltar, not by encircling the Mediterranean. The authors of publications emphasize that this concept is also confirmed by similar tool stones discovered on the Iberian Peninsula and in the north of the African continent, as well as the presence of the remains of African fauna in the south of the Peninsula Iberian, such as the remains of a hippopotamus found in Ors or Therapithecus bones Oswaldi, an African chief of a baboon, found in Cave of Victoria near Murcia. Researchers also emphasize that in no other place on an alternative route from Africa to Europe, no elders were found. The evidence. The route through the Strait of Gibraltar now requires to feet about 14 kilometers across the sea, but as the researchers suggest, perhaps in the past was shorter at certain times due to the high tectonical activity in this region and sea level fluctuations conducive to migration. Scientists also point out that they have identified migrations of African fauna through the Strait of Gibraltar in earlier times between 6.2 and 5.5 million years ago, when the Strait of Gibraltar was very narrow. Scientists flew into the eye of Hurricane Beryl. All of it, they recorded on the video. The National Agency for Affairs, Oceans and Atmospheres, NOAA, sent its specially prepared aircraft straight into the eye of the Hurricane Beryl. All to gather valuable information about the hurricane and improve forecasts for its route and intensity. The first hurricanes in the Atlantic usually form at the beginning or in mid-August. But Hurricane Beryl, which hit the month ago the coast of the United States, appeared in June. In the beginning, it looked like an ordinary tropical storm, but in a short time it grew into the 5th category on the Saffir-Simpson scale, which means that the winds reached a speed of 260 km per hour. The hurricane has already weakened and is now classified as the tropical depression. According to the researchers, the early appearance of a strong hurricane is related to climate change and rising temperatures, both air, as well as ocean waters. Hurricane Beryl, even before hitting Texas, aroused disputes, anxiety. It appeared earlier than usual, which is why the researchers acknowledged that he can have more surprises in his own place. To check it out, they directed a special plane straight to Beryl's eye. Planes were sent into the hurricane eye for the monitoring. NOAA uses such data to forecast routes and the intensity of the hurricanes. Of course, such flights are dangerous and are associated with a big risk. Below is a recording of the recent meeting with Beryl. In the eye of the hurricane, i.e. at its central point, the conditions are very calm compared to hell raging outside of it. There is no rainfall, the wind is very weak. Above the observer, the sky is cloudless, which is perfect. You can see the recordings. In the eye there is a higher temperature and lower pressure in compared to the conditions outside. NOAA regularly flies in hurricanes to make them monitor and predict their development. The agency has a special test cell hurricanes called NOAA Hurricane Hunters. It has special aircraft that play an important role in research and strength forecasting the hurricanes. These machines are actually flying weather stations. The data collected by them helps to better understand the storm processes, improving the prognostic models. NOAA has 2 such aircraft, Kermit and Miss Piggy. Each of these aircraft can stay in the air for up to 12 hours. They have on board scientific instruments that are dropped from a height together with the wind, transmitting valuable data on wind speed, humidity, air pressure, temperature, or storm structure, and in the position of the hurricane, of course. They also study the ocean temperature by checking whether it does not fuel the force of the element. The aircraft also have radars on the tail and in the lower part hull that scans hurricane vertically and horizontally, giving scientists and prognosticians image in real time. The Higgs boson seems to not contain any traces of new physics. Although the discovery of the Higgs boson was announced in 2012, physicists still did not know its properties with satisfactory accuracy. What the truth? After 12 years of research we know a bit more about this particle, but it looks like that it does not contain elements of new physics outside the standard model. Dick physicist Peter Higgs, who died in April in 1964, he proposed a new particle. He said the theory that there must be a particle, a subatomic, that explains how other particles gain mass. In 2012, the year in the Large Hadron Collider, LHC, managed to detect such a particle that they began to be called the Higgs boson or divine particle. Since his discovery, physicists have been trying as accurately as possible learn the properties of this very important elementary particle. The task is extremely difficult both for experimental challenges and numerous obstacles of computational. It is believed that the discovery of the Higgs boson was the largest discovery made in the Large Hadron Collider. Award-winning achievement, he explains, among other things, the existence of mass. The Higgs boson is a fundamental element of the model standard, a set of theories describing the basic elements of construction the universe. The Standard Model is one of the most important theories in modern physics. It is a theoretical framework describing nature most the basic level. Scientists use it to describe all known elementary particles in the universe and forces through which they interact. The model began to be developed in the 1970s. However, scientists are aware that it is limited, and although it is ours the most comprehensive and accurate model for particle physics elementary, contains gaps, so it is not able to explain everything, for example, gravity or what dark matter consists of or why there is much more material in the universe than antimatter. Scientists are looking for particles that behave in a different way than you would expect from the predictions of the Standard Model to be able to explain some of these mysteries. They hoped it was the Higgs boson. It will allow you to extend the Standard Model. But this part turned out to be so difficult to generate and observe that despite 12 years of research, its properties they have not yet been known with satisfactory accuracy. In recently published works by a group of physicists from the Institute Nuclear Physics of the Polish Academy of Sciences, IFJ Pan, in Krakow, RWTH Aachen University and the Max Planck Institute Physik, MPI, from Garching near Munich, the properties of the Higgs boson are described slightly more accurately. These findings suggest that within the studied mechanisms responsible for the formation of Higgs bosons you can see the trailers of the new physics. Results and description of the research appeared in the journal Physical Review Letters / For physics, one of the most important parameters related with each elementary or nuclear particle is an active cross-section for specific the collision. It does not know how often we can expect the appearance of a given particle in collisions of the selected type. We have concentrated on the theoretical determination of the cross-section value of Higgs active bosons in colliding of gluon-gluon. They are responsible for production of about 90%. Higgs, whose traces of presence have been recorded in LHC accelerator detectors, explains in a message published on the IFJ-PA website Dr. René Poncelet. The essence of our work was the desire to include with the designation of the active cross-section on the production of Higgs bosons certain amendments, which, due to the seemingly small contribution, are usually overlooked, because they ignoring significantly simplifies the calculation. For the first time, we succeeded to overcome mathematical difficulties and set these amendments, emphasizes Professor Michael A. Koch from RWTH. On how important the role of higher row corrections is for an understanding of the properties of Higgs bosons may be evidenced by the fact that the ones calculated in article secondary amendments, seemingly small, contribute almost a fifth to the value of the searched active cross-section. For comparison, amendments the third order is at the level of 3%, but they reduce uncertainty computation to just 1%. A novelty in this article was taking into account the influence of masses of beautiful quarks, which led to a small but noticeable additional transfer of value by about 1%. It's worth it in this place reminds that the LHC collides protons, i.e. particles consisting of two upper and one lower quark. Momentary presence in the interior protons of larger masses such as beautiful quark is a consequence the quantum nature of strong interactions binding quarks in the proton. We read in the communication. Poncelet adds that the values of the active cross-section found and those measured in the LHC are actually the same. With the agreement, of course, inaccuracies of computational and measurement. So it seems that within the mechanisms we studied for the formation of Higgs bosons there are no trailers for new physics, at least for now, he concludes. In the communication of the IFJ-PAN it was noted that these findings are not they exclude new physics in the phenomena associated with the birth of the Higgs bosons. The results of experiments from the fourth research cycle of the GREAT are waiting for the analysis the hadron collapse, and they can change a lot. More and more observations new particle collisions may allow to narrow down the measurement uncertainties so that measured range of permitted active cross-section values for production Higgs bosons will stop being matched with the theory. Night brands have better results in cognitive tests than the wounded birds. Recently published research suggests that there is relationship between a person's preference for morning or evening activity and the performance of the brain. Scientists say people declaring themselves night brands usually have higher scores in cognitive tests. Scientists from Imperial College London analysed data more than 26,000 people to learn how different aspects of sleep, including duration, its patterns, and quality, affect the sharpness of the mind and the cognitive ability. Studies have shown that people who prefer evening-night the lifestyle has better cognitive functions than people starting from activity the dawn. Results and description of the research appeared in the journal BMJ Public Health. The data the researchers worked on came from the UK Biobank, an extensive database maintained since 2006 and focused on genetic predispositions and environmental factors, such as the way nutrition or lifestyle, for the development of the disease. The researchers analyzed information about adults who revealed what time of day they felt the most vigilant and productive and who have completed a number of cognitive tests. They also checked their sleep patterns and other factors. Analyses have shown that sleeping 7 to 9 hours it was optimal for the brain every day, increasing cognitive functions such as memory, reasoning, and the speed of information processing. Sleeping less than 7 hours or more than 9 hours had a distinctly detrimental effect on functioning of the brain. Scientists have also determined that the chronotype of a person, i.e. individual preferences as to evening or morning activity, also affected for the test results. Night brands, i.e. people who are naturally more likely active in the evenings, performed better in cognitive tests compared with morning birds, i.e. people focused on activity from the very morning. The analysis has been adapted to other health factors and lifestyle such as age, sex, smoking, and alcohol consumption, and the presence of chronic diseases such as heart disease and diabetes. The younger ones, people without chronic diseases, generally obtained higher scores. A healthier lifestyle was also associated with better cognitive performance. Our study found that adults who are naturally more active in the evening tended to better results in tests cognitive than people who have been active in the morning. These chronotypes can affect our cognitive roles, said Dr. Roha West of Imperial College London, the lead author of the study. This does not mean that all the wound birds have inferior cognitive functions. The results reflect the overall trend, she added. West also emphasized that it is important to remember to sleep not too long or too short. This is crucial for keeping the brain healthy and for its proper functioning. However, although the length of sleep was important in research, people who reported insomnia problems did not get significantly lower results in cognitive performance. This may indicate that you should take considering specific aspects of insomnia, such as its severity and time the duration. Sleep duration has a direct effect on brain function. We believe that proactive management of sleep patterns is really important for strengthen and protect the way our brains work. Daking Ma of Imperial College London, CO author of the paper. Not all experts agree with those made in publication findings. Without a detailed picture of what is happening in the brain, we don't know if being a wound person or evening affects memory and thinking, or a decrease in cognitive functions causes changes in sleep patterns, admitted Jackie Hanley from Alzheimer's Research UK. Jessica Chelowakis from Brunel University London added that the study had significant limitations because it did not take into account education or time of day in which cognitive tests were conducted to the UK-based The Biobank. Butter produced from carbon dioxide. Backed by Bill Gates, the startup has developed a way production of carbon dioxide fats taken from the air and hydrogen from the water. In this process, there is no need to use animals, plants, or the agricultural land. According to former head of Microsoft, butter made with the use this innovative technology tastes as good as traditional ones produced by the methods. California startup Savor, backed by billionaires with Microsoft, Bill Gates, says he has developed a way to make fats without the participation of animals or plants. Alphault of carbon dioxide from the atmosphere and hydrogen taken from the water. Agriculture accounts for a significant part of global emissions, the greenhouse gas. These products can reduce emissions from this branch the economies. This process does not release any greenhouse gases, no uses agricultural land and uses less than 1,000 water in compared to traditional agriculture, Gates explained in a blog post. One of the newest Savor products is butter, which according to Bill, it tastes like real. It tastes really good. How real? Because it is chemically so. I couldn't believe I wasn't eating a real butter, the billionaire said. Synthetic production of food fats with the help of chemical and biological processes have considerable potential. Raw materials for this method are the same as those used by plants, hydrogen taken from water and carbon dioxide from the air. The process is based on the concept presented last year in Nature Sustainability by University of Scientists California Irvine. Researchers have shown that fats from animals producers produce about 1 to 3 grams of carbon dioxide per thousand the calories. But you can produce the same amount of fats in the lab at emissions less than a gram. Large-scale synthesis of edible particles with the help of chemical and biological agents without agricultural raw materials is very real. The possibility. This method can avoid huge amounts of gas emissions, greening and at the same time protecting biodiversity in areas that they could be grubbed up for agriculture, Stephen Davis said last year from UC Irvine, lead author of the study. Fats are made up of different chains of carbon atoms and its hydrogen. These chemical building blocks can be obtained from water and air, and then use biochemical processes to convert them into fats, which are molecularly identical to those found in animals and plants. New type of fat that production does not require participation animals or large areas of the earth, is produced in a thermochemical system closer fossil fuel processing than food production. In this process you can build fat molecules to form carbon dioxide, hydrogen, and carbon chains the oxygen. Researchers have developed a process of downloading carbon dioxide from air and hydrogen from water, heating, and oxidation, which it causes the separation of fatty acids and then the formation of fat. The result is real fat particles like the ones we get from milk, cheese, beef, and vegetable oils, Gates wrote on his blog. Kathleen Alexander, Savor's chief executive, admitted that the company's products can be sold in stores in a few years. We are currently on pre-commercial stage and work to obtain consent from the authorities regulatory to be able to sell our butter, she said. The startup also has in plans to develop similar methods of production of cheese, milk, or ice cream. The question is whether such butter and other products made they will synthetically gain recognition in the eyes of customers. Incentive people to resign from your favorite dairy and meat products for more experimental food products can be a challenge. Blocking one of the proteins has increased the life of the mouse by more than 20%. Blocking a protein that promotes inflammation has extended a mouse's life of more than 20%. Moreover, inhibition of interleukin-11, as we are talking about it, the incidence of cancer rods has also decreased in other age-related health problems. This research gives hope for development of effective anti-aging therapies. Scientists from the Medical Research Council Laboratory of Medical Science and Imperial College London, in collaboration with researchers from Duke News Medical School in Singapore, found that blocking a molecule known as interleukin-11, IL-11, may extend the life of mice, suggesting that IL-11 inhibitors they can also have anti-aging effects in humans because these proteins produced they are also through our bodies. Candidates for IL-11 blocking drugs are now tested in humans in anti-cancer therapy studies. Results and description of the research appeared in the journal Nature. IL-11 belongs to the cytokines, the proteins involved the immune response. These proteins have broad and complex functions. They can initiate inflammation, but also counteract it. They stimulate the processes immune, stimulating the cells of the immune system to react. So far, more than 100 such proteins have been described and new ones are still being discovered. Together they create a complex and extremely sensitive system. Scientists study IL-11 from many years. In 2018, they showed that IL-11 is a prophylactic protein and pro-inflammatory, refuting years of misconduct as anti-fibrotic, and it's the opposite. Chronic inflammation contributes to related diseases with aging. As the body ages and accumulate damaged proteins and other molecules, the immune system often sees them as signs of possible infection. This may cause inflammatory responses that may cause further damage and contribute to cancer or autoimmune diseases. The link between IL-11 and aging processes has been discovered by accident. Anissa Rajajija from Duke National University of Singapore Medical School tested the IL-11 detection method. Coincidentally, she included a sample in the test's proteins taken from the old rat. The test showed that IL-11 level was significantly higher in this sample than in samples from younger rats. This project started in 2017, when one of our collaborators sent us some tissue samples for another project. Out of curiosity, I have done several experiments to check the IL-11 level. From the readings it was clear that the IL-11 level increased with age, said Wijija. Researcher admitted rising IL-11 levels contribute to negative effects in the body, such as chronic inflammation, problems with wound healing or regeneration of organs after injury. She also added that although research has been conducted in mice, the team hopes their findings they will have an impact on people's health and lives. These studies are an important step towards a better understanding of aging processes. We tried on mouse therapy that could potentially prolong healthy aging and reduce healthy aging, the physiological symptoms of this process, she added. By checking IL-11, the researchers examined different samples. It was taken from young and older mice. IL-11 was much higher in older individuals, in skeletal muscle, fat, and liver tissue. The next step was created by mice devoid of a gene that produces interleukin. It is up to 5. It allowed rodents to live longer by an average of more than 20% compared to mice with normal level of IL-11. The team scored similar results when they used for 25 weeks IL-11 neutralizing antibody in mice at 75 weeks of age. That's more or less the equivalent of 55 years in man. In mice given an antibody from 75 a week of age to death, the life expectancy increased by 22.4%. Males and 25% in females. Mice lived an average of 155 weeks, compared to approximately 120 weeks in control mice that have not received an antibody. In the following recording on the left you can see the mice given the antibody neutralizing IL-11, on the right, normally aging. Both groups, the rodents in the film, are 90 to 95 weeks. Moreover, among the rodents given antibodies, there was much fewer cases of cancer and diseases caused by fibrosis, chronic inflammation, and weakened metabolism, which are features characteristic of the aging process. For this, researchers they observed few side effects. These discoveries are very exciting. The treated mice had fewer cancers and no typical signs of aging. We have observed also, reduce muscle atrophy and improve their strength. In other words, the old mice receiving IL-11 neutralizing antibody were healthier. Although at least the findings concern mice, there is a tantalizing possibility that neutralizing drugs IL-11 can have a similar effect in humans. Anti-IL-11 therapies are currently being checked in human clinical trials for other conditions, which potentially for exciting opportunities to explore their impact on aging the body in the future, said Stuart Cook of Imperial College London, co-author of the publication. Life on Venus? Astronomers have rediscovered the phosphine on the neighboring planet. They also found ammonia. In 2020, the world was announced about the discovery in the atmosphere of Venus traces of phosphine. This gas is produced on Earth by some anaerobic bacteria that triggered speculation about life on the neighboring planet. However, over time, more and more scientists began to question the regularity sensational findings, indicating a data error. Now scientists from 2 independent bands have found the phosphine in the clouds of Venus. They also found traces of ammonia. Scientists from 2 independent teams have reported discovery in the hot atmosphere of Venus phosphine and ammonia. Both of these gases are referred to as biomarkers, which may suggest that there are any exist in the clouds of Venus some kind of life forms. Discoveries were reported a few days ago at the National Astronomical Meetings at the University of Hull, UK. Venus is often called Earth's sister or planet the twin one. This is due to the size comparable to the Earth, because the conditions there the prevailing are far from the earthly. The temperature on the surface of Venus is about 500 St Celsius. Venus is the closest planet to Earth and revolves around own axis in the opposite direction than most planets in the system. It's a sunny one. The planet has a very dense atmosphere and the pressure there is 92. It's often bigger than on our planet. The idea of searching for microbiological life on Venus it's a new one. Some models suggest that Venus had a climate suitable for occurrence liquid water on the surface for up to 2 billion years. But it's more about the atmosphere of the twin planet, not its surface. Earth's terrestrial microorganisms are able to get into the atmosphere. They were found alive at heights up to 41 kilometers by scientists using specially equipped balloons. It shows that life may be present in the atmosphere. As for the hellish conditions prevailing on Venus, this is an ever wider catalogue of earthly microbes known to be found. They live extremely difficult environments, including hot springs in Yellowstone, area of hydrothermal chimneys on the ocean floor, toxic sludge in polluted areas or acidic lakes around the world. The idea that microorganisms can live in the clouds of Venus as the first was advanced by astronomer Carl Sagan and biophysicist Harold Morowitz in 1967. A few decades later, David Grinspoon, Mark Bullock and their colleagues expanded this idea. To check this towards the twin planet, a series was directed probe that showed that temperature and pressure in the lower and middle Venus atmosphere, at an altitude of 40 to 60 kilometers does not exclude life the microorganisms. On the surface of the planet it would be impossible because of temperatures of nearly 500 degrees Celsius. But at an altitude of 40 to 60 kilometers above the surface, the temperature and pressure are closer to the earthly conditions and potentially, they could allow some very resilient extremophiles to survive. The first reports of the phosphine in Venus appeared in 2020 and immediately caused a sensation. However, since the announcement of the results this finding was questioned and experts pointed to errors in research. At the meeting of astronomers at the University of Hull, researchers from Imperial College London announces rediscovery in the clouds of Venus phosphines. Phosphoro, a gas also called a phosphine, is a compound that on Earth it is produced, outside the laboratories, by anaerobic bacteria. The relationship it is also caused by volcanic activity. Astronomers from the University of Cardiff reported that in the atmosphere of the neighbouring planet was also found by ammonia. It's a gas that's left again. The first one on Venus was detected in the 1970s. On Earth it is produced mainly through biological activity, but also industrial processes. His being on Venus is hard to explain. Scientists know no process chemical on Venus that could produce this compound. The presence of these two gases in the Venusian atmosphere does not. It is irrefutable proof of the existence of a form of microbial life there. However, it ignites the imagination. If Venus went through a warm, damp phase in the past, with global warming, would evolve to survive in the only niche he had, the clouds, Dr. Dave said at the meeting. Clements from Imperial College London. New observations of Clement's team headed by Clements, the use of the James Clerk Maxwell JCMT telescope in Hawaii showed the existence of a phosphist in the atmosphere of Venus. Following her signature, the scientists saw that the phosphine seems to follow the cycle of day and night on the twin planet. Our findings suggest that when the atmosphere is bathed in sunlight, phosphap is destroyed. All we can say is that the phosphine there it is. We don't know what makes it. Maybe it's a chemistry we don't understand. Or more than a life, Clements said. There are many quirks in Venus' atmosphere. Phosphors of the North, he's just another one of them. Among other strange things is the way the amount of water and sulfur dioxide in the atmosphere changes over time. We do not know why, added Clements. The same with ammonia. Astronomers emphasize that it is difficult to explain its presence in the clouds of Venus with the help of known phenomena, atmospheric or geological. His traces were discovered by researchers from the University of Cardiff with the Green Bank telescope. They suggested if on Venus there is life, it can use ammonia to neutralize sulfuric acid, from which consist of Venusian clouds, thanks to which small water droplets are made. They would be habitable. If there are microbes in the clouds of Venus, they can produce some gases we wouldn't expect. And here comes the ammonia, because it can be used as a way to neutralize acid, Professor Jane Greaves of the University of Cardiff said. Greaves has headed a team of researchers who caused a sensation in 2020 with references about phosphonates on Venus. These are very exciting discoveries, but it should be emphasized that these are preliminary results and the need for further work to learn more about the presence of these two potential biomarkers in the clouds of Venus. Nevertheless, however, it is fascinating to think that these findings may indicate possible signs of life or some unknown chemical processes. It will be interesting to see what the research will reveal in the coming months and years, said Robert Massey of the Royal Astronomical Society. Spacesuits inspired by Diuna, they'll turn urine into drinking water. Filtration system installed in wearable suits by the protagonists of Frank's cult science fiction novel Dune. Herbert, who turns urine into drinking water, inspired scientists to develop a similar solution. Such a suit could allow astronauts performing long spacewalks and provide greater comfort and hygiene than the current solution. Stealing physiological needs in space is enough, it's a burdensome task. On board the International Space Station, ISS, fade and the urine is sucked. Gentlemen, for the latter need, they can use special, personal funnel connected to the pipe with a small suction force. But during spacewalks, astronauts take out their needs in the suits. It is inconvenient and unsanitary, and even waste, because urinary water is not recovered, and in space all the resources are extremely valuable. The solution to these problems would be suits, such as these, the novels worn by the characters of the novel Diuna, which absorbed and purified water lost during sweating and urinating and then processing it into the drinking water. This fantasy can become a reality. Scientists from Cornell University in New York recently unveiled a prototype of an innovative system collection and filtering of urine in spacesuits. Project details were published in the journal Frontiers in Space Technology. Urine and sweat are already recycled on board the ISS. Water is recovered from them, which is used to generate oxygen. However, the equipment with which it can be made is large and bulky, and it's not in a spacesuit. It's different when walking the cosmic. Astronauts carry a liter of water and are equipped with the MAG system, maximum absorbency garment, which, apart from the fictitious name, boils down properly to a large diaper. After such a walk, the diapers go to the tank from the waste that is shed into the atmosphere, burning. MAG is known in the astronaut's environment. He was introduced in the 1970s. It's a diaper, only made of super-absorbing material. Complaints about this solution are many. Some of it, astronauts even limit food and drink intake before walking space to limit the use of MAG. Besides being uncomfortable, it often leaks, which can lead to urinary tract infections. MAG reportedly leaked and caused health problems, such as urinary tract infections and gastrointestinal problems. In addition, astronauts currently only have a liter of water in the beverage bags in their own suits. This is not enough for longer walks on the moon, which they can last up to 24 hours in an emergency, Sophia Etlin said. Cornell University, co-author of the study. Chris Mason of Cornell University says its current the solution is good for spacewalks that only last a few hours, but increasing activity in space means a better solution will be needed. Mason and his colleagues developed a device the size of a shoebox that can urinate its recycling. The prototype collects urine, cleanses it and can return it. The astronauts are drinking water in 5 minutes. The creators of the suit they hope it can be implemented before the end of the decade by NASA and Artemis program partners. The proposed suit system consists of a container of molded silicone with different shape and size for women and men. It is found in underwear made of many layers of elastic material. Silicone vessel connects to activated vacuum pump moisture that automatically turns on as soon as the astronaut starts giving back the urine. After collection, the urine is directed to the filtration system where it is recycled with an efficiency of 87%. The system uses the phenomenon osmosis for urinary water removal, as well as a pump to separate water from salt. According to the authors, collecting and cleansing half a liter of urine it only takes 5 minutes. What's more, the purified water can be enriched with electrolytes and a subordinate as an energy drink. The system has dimensions of 38 cm by 23 cm by 23 cm and weighs about 8 pounds. It was considered to be compact and light enough that it can be moved on the back of the suit. As part of the tests, for the autumn, 100 people from New York will be checked the system for convenience and functionality. An unknown source of oxygen discovered at the Pacific. New research has shown that conglomerates spread on the ocean floor different minerals the size of a potato called polymetallic concretions. They produce oxygen. This process runs at a depth of 4,000 meters below the surface, in total darkness and without the help of living organisms, which undermines our knowledge about the development of life on Earth. Formations called polymetallic concretions or concretions manganese are pieces of rock consisting of different elements. They are present on the bottom of the oceans at a depth of 4,000 to 6,000 meters below the surface. These lumps, about sizes from tiny particles to the size of the average potato, contain large amounts of manganese, iron, nickel, cobalt, copper, and rare earth elements, which is why they were considered as a source of raw materials. But they are a source of oxygen. This unexpected discovery undermines the widespread belief that for the production of oxygen needed for breathing photosynthetic organisms are needed. Results and description of the research appeared in the journal Nature the Geoscience. Spontaneous reaction quietly rolling in the dark depths oceanic, discreetly produces oxygen, without the participation of life. The discovery of the dark oxygen, as scientists called oxygen produced at the bottom of the oceans, undermines long-sustained assumptions that they are photosynthetic organisms, such as plants and algae produce oxygen on Earth. In order for oxygen life to be created on the planet, it must be oxygen, and to our knowledge, the generation of oxygen on Earth began with next synthetic organisms, said Andrew Sweetman of Scottish Association for Marine Science, SAMS. But now we know that oxygen is produced in the sea depths where there is no light. I think we have to ask yourself again about where life could begin. Oxygen? He added. Polymetallic nodules that produce this oxygen contain metals such as cobalt, nickel, copper, lithium, and manganese, which are critical the elements used in batteries, said Franz Geiger from Northwestern University, co-author of the study. Several large mining companies have currently, to extract these valuable elements from the seabed from depths from 4,000 to 6,000 meters below the surface. We have to think about how extract these materials so as not to exhaust the source of oxygen for deep sea life, he added. The discovery was made during a research expedition on a ship on Pacific in the so-called Clarion-Clipperton area of about 3,000 miles west of Mexico. Oxygen was detected during the intake of bottom samples. Initially, the team of researchers assumed they have broken equipment. When we first received the data, we thought the sensors are flawed, Sweetman said, adding that no studies so far they did not report the production of oxygen in the depths of the sea. Researchers used another the method, but this one also pointed to the detection of oxygen. When both methods gave it the result itself, we knew that we were dealing with something groundbreaking, he emphasized. Scientists wondered where the oxygen detected came from. Geiger in a study from a few years ago found that rust in combination with salt water it can generate electricity. This prompted scholars to consider whether the concretions polymetallic at the bottom produce enough electricity, to produce oxygen. This chemical reaction is part of the water electrolysis process C, i.e. water distribution to hydrogen and oxygen with energy the electric one. To check this, some taken from polymetallic bottoms the lump was sent to the Geiger lab at Northwestern University. Studies have shown that a voltage of only 1.5 volts is enough, which is the same, what a typical AA battery has to split seawater. On the surface single concretions, the researchers recorded a voltage of about 0.95 V, but when many the concretion is focused together, the tension can be much greater, as in case of batteries connected in series. It seems we've discovered a natural geobatic. Those geobatteries are the basis for a possible explanation for the production of dark oxygen in the ocean, Geiger said. Scientists agree that the mining industry should consider this discovery before planning deep mining activities the sea. According to Geiger, the total mass of polymetallic concretion in the zone itself Clarion-Clipperton is enough to meet the world's demand a decade. However, the scientist emphasizes that mining is associated with degradation the environment. In 2016 and 2017, marine biologists visited places in which minerals were mined in the 1980s. The 20th century and discovered that in these areas there are not even bacteria back, Geiger said. There was no mining of marine life flourished. Still no one knows why such dead zones have persisted for decades. However, it puts it a big question mark for mining, because the variety of fauna at the bottom oceanic and areas rich in concretions is higher than in the most various tropical rainforests, he added. Accidental discovery of the Curiosity rover. The rocks it is made of pure sulfur. NASA has announced an important discovery on Mars. The rover and riveter Curiosity moving through the region of Mount Sharp accidentally split the rock he was driving on. It turned out that it is made of pure sulfur. On May 30th, the Curiosity rover studied hollow and steep slopes Mount Sharp Canyon Geddes Vallis. During the trip accidentally invaded the small stone, which under the influence of the weight of the rover, weighing about 900 kilograms, it just broke. As the rover's cameras focused on what the rover stumbled over, scientists have spotted yellow crystals of pure sulfur gloss among fresh exposed intestines of rock. Sulfur has already been detected on Mars, but only in combined with other elements in compounds known as sulfates. To this the pores of pure sulfur were never found on the red planet. Scientists they suspected that somewhere on Mars, pure sulfur was certainly found, but they did not expect to find it in the surface rocks. What's more, in Geddes Vallis sulfur canyons is quite a lot near the rover is whole a field of bright rocks similar to those that Curiosity accidentally split up. Finding rocks made up of pure sulfur is how to find an oasis in the desert, Ashvin Basaveda of Jet Set Propulsion Laboratory. It shouldn't be there, so now we have to explain it. Discovering strange and unexpected things makes exploring the planets is so exciting, he added. NASA has been sent to Mars by the Curiosity rover since 2012. Traverses the wasteland of the red planet, drilling in the rocks and passing through samples taken by an advanced chemical laboratory that it has on its aboard. Its main purpose is to seek evidence of the existence of an ancient life. The life. Curiosity landed in southwards from the equator the crater of Gale. The landing place was selected based on the photos satellite that has shown that probably in the distant past there was liquid water. Mount Sharp lies in the center of the crater and the rover is climbing for it since 2014. The mountain is crossed by the Geddes Vallis Canyon. It was an area interest of NASA's scientists long before the Rizomrazi landed the red planet. Scientists believe the canyon was hollowed out by the flowing water that took smaller and larger boulders with it. According to them, each the mountain layer represents another period in the history of Mars. There was an exciting amount of activity here. We are looking at multiple flows down the canyon, including vigorous floods and flows rich in boulders, said Becky Williams of the Planetary Science Institute in Tucson in Arizona. Although sulfates are quite common on Mars, this is the first time that sulfur in its pure form has been found in the neighboring the planet. Sulfate is salts or esters that form when sulfur, usually in the form of the compounds, is mixed with other minerals in water. When the water evaporates, the minerals mix and dry, leaving the sulfates. These minerals sulfate can tell us a lot about Mars. for example, about its history, water, and how it was weathering over time. In turn, pure sulfur is created only in a narrow range the conditions. Researchers did not think they may have occurred in the study by the Curiosity rover region of Mars. Importantly, sulfur on Earth is essential an element for all life. It is usually taken by organisms in forms of sulfate and used for the production of essential amino acids, which living organisms need to make proteins. Researchers are now going to determine, based on what we know of Mars, how the sulfur was found in Giedi's Vallis. It will take a little time. Scientists are likely to have to model evolution in detail geological of the red planet. In the meantime, Curiosity will continue. He explored the wilderness of the red planet. Currently moving along canyon to see if any other surprises are lurk for another. It's a rock. Aboriginal heritage 47,000 years ago destroyed by a mining company. In 2020, 2 holidays for Aboriginal caves were blown up Pilbara region of Western Australia. The responsible company for it, Rio Tinto, wanted access to huge resources in this way, or iron. We will never know what has been destroyed, but he has just shown a report on the archaeological significance of the caves, which shows that the Aborigines used them 47,000 years ago. Rio Tinto, one of the biggest companies in May 2020 excavation in the world, he blows up 2 sacred caves for Aboriginal people located in the Pilbara region of Western Australia. The company has obtained to Australian authorities all necessary permits in 2013. A year later archaeologists have arrived at the site. They found thousands of artifacts from the last 28,000 of years, but the oldest evidence for human presence in caves dates back to 46,000 years back. In new research, the researchers pushed the date of the presence of man in the caves another 1,000 years ago. It seems that Aboriginal people first used grottos about 47,000 years ago, possibly in the last ice age, up to just a few decades before destruction of the cave. Results and description of the research appeared in the journal Quaternary Science Reviews. Archaeologists have begun excavations in places named Juukan Gorge 1 and 2 in 2014, 6 years before blowing up them in the air. They were accompanied by Aboriginal representatives. They found then thousands of artifacts, including an ancient tail from human hair and tools, and dozens of animal bones. The results of the excavations led to making last-minute attempts to stop work on this. The area, however, was unsuccessful. Places of excavations were in a gorge filled with caves, about 60 kilometers northwest of Tom Price, in the region Pilbara in Western Australia. Juukan 1 and 2 were part of the caves that make up the system. Researchers indicate that once a rivet sailed through a rive gorge the river at some times of the year, and in others the gorge was dry. He is now finding it will be about 500 kilometers off the coast, but 10,000 years ago, when the sea level was lower, it was almost 1,000 kilometers from the coast. Now the area is part of the Rio Tinto iron ore mine. This gorge is extremely important for the Karema people. The place it is also one of the oldest known places of Aboriginal settlement in Australia. During the excavations, scientists found thousands of artifacts there. Many of them were wearing traces of resin made of grass from the genus Spinifex, which was probably used as a kind of glue to connect tool parts or the guns. Researchers also found numerous animal bones, including kangaroos, emu and even Australian spikes. One of the Burka's tibias before about 30,000 years was covered in a cobra, indicating a ritual function. The aforementioned braid from the human hair. Dating has shown that the person to whom the hair belonged lived about 3,000 years ago. The hair was tested for DNA, and the results indicated that she was related to the Aborigines of the Karema people. After the destruction of the Juukan 2 site, archaeologists returned to the place. They removed about 150 cubic meters of rubble, which used to be the roof and the back wall of the cave. Under the rubble, they found traces of organic material, and then the remains of the cave floor. More was found during the work: braids, beads from shells, which were probably brought from coasts, and fragments of the Tasmanian devil's jaw, an animal that died out in mainland Australia over 3,000 years ago. There were also blades from bones, grindstones and artifacts from the clogged stone. Even traces of starch on stones have been discovered. Artifacts, which shows that the processing of the plant Ipomoea caustata had there was a place there 42,000 years ago. The Uecan 1 and Jukan 2 were the only known places that contained evidence of the continued presence of humans during the last the Ice Age, which was not near the coast. The tongue of elephants. Scientists have registered on the video. What is the command "Let's go"? Scientists have documented for the first time on the video, as elephants use infrasound hums to signal to their group that it's time to move on. Registered calls are initiated by dominant the males. Until now, it was believed that such behaviors are the domain of females. An international team of scientists has shown how males African elephants signal that it's time to get on the road. The command "Let's go" he initiates a dominant male, making a deep murmur. In turn, they respond the other elephants, and their voices overlap with a loud, an infradnib require that carries through the savanna. The findings are described in the journal Peer-J. Scientists documented the signaling command on the recording departing from the waterhole in Etosha National Park in Namibia. It has been initiated by the male. Until now, researchers thought that in compact social groups elephants of this type of behavior are a feature of females. We were amazed when we discovered that male elephants, which he usually considers himself, lose social ties. They engage in this sophisticated vocal coordination to induce action, said lead author Caitlin O'Connell-Rodwell's research from Stanford University. These sounds they show us that there is so much more going on in their vocal communication than previously, she added. O'Connell-Rodwell first recorded a murmur of "Let Go" issued by males in 2004 during night field studies. In the years 2005 to 2017, together with colleagues from other institutions, collected data at Musher Waterhole in Etosha National Park, mainly in the dry season. Scientists they used high-tech recording equipment, including varied microphones and night vision video cameras to capture behavior elephants and infrasound vocalizations, inaudible to the human ear. Scientists analyzed vocalizations for properties, acoustic and patterns. In the analysis, they paid attention to which elephants they initiated murmurs to understand relationships and hierarchy among males. They also checked how other individuals responded and the sequence of events leading to the coordinated activities. The command "Let Go" issued by male elephants is very much similar to the same command issued by females. O'Connell-Rodwell and the team hypothesized that male elephants are probably learning this behavior when they are young. They grew up in a family where all female leaders they were involved in this ritual. We believe that as you mature and create their own groups, adapt and use these learned behaviors to coordinate with other males, O'Connell-Rodwell said. After the command "Let's Go" issued by the initiator, the rumbling of another individual, with each elephant waiting for the previous sound, it will end before he adds his own. It creates a harmonious, alternating pattern resembling a quartet of soloists. This study is a continuation of another one. Used artificial intelligence to reveal that wild elephants African have unique names for themselves, which indicates they use nouns in their communication. In our article we show that elephants they use verbs in the form of a rumbling. If they use combinations noun time together, this is the syntax. It's a language, she said. O'Connell-Rodwell. In addition to linguistic insights, the study also reveals that some dominant male elephants play key roles in their group's social, helping to maintain consistency and stability. These individuals take roles of mentors. They care about young people who always want to have physical contact. Older people males are ready to take them under their wings, lead them, share with them resource and participate in their emotional ups and downs, she explained. O'Connell-Rodwell. Our findings not only highlight complexity and wealth the social life of male elephants, but also deepens our knowledge of the subject, how they use vocalizations in rituals and coordination, she pointed out the researcher. Advanced cognitive abilities of bats. They have a good memory and can plan ahead. Scientists from Israel have studied the cognitive abilities of the living in the wild, bats. It turned out that not only have a good memory, but they can also plan ahead and show self-control in their own behavior. The results challenge the traditional notion that cognitive abilities, such how episodic memory and future-oriented behavior, are exclusive a feature of people. Scientists from Israel have discovered that bats can bringing in past experiences and planning the future, indicating presence of episodic memory and the ability to travel mentally in time. What was more, researchers have determined that these flying mammals can plan within advance and deny yourself the benefits to receive later. It's a bigger reward. Results and description of the research appeared in the journal Current Biology. The research focused on the colonies of Nile rally, bats found in Africa and the Middle East, feeding mainly with fruit. Researchers equipped the individuals from the colonies with GPS locators so you can track their routes and preying patterns. Observed bats are inhabiting although the zoo maintained by Tel Aviv University, but they were allowed to leave the colony, which allowed them to monitor the animals in with their natural environment. For many years, cognitive abilities to remember personal experiences, authentic memoir, and planning in advance considered the exclusive feature of people. However, more and more research suggests that different animals also have such abilities, but almost all of these studies it was carried out in laboratory conditions because field studies on these issues are difficult to carry out. Trying to test these abilities in animals living in the wild, we designed a unique experiment consisting in a colony of free-living Nile redheads from the Garden of Research Zoological to them. N.I. Meyer Seagulls at Tel Aviv University, said. Leading the research Professor Yossi Yovel from Tel Aviv University. Scientists have assumed that bats that feed on fruit, they had to develop the ability to track the availability of food both spatially, where are the fruit trees, as well as in time, or when the trees they are bearing fruit. Moving through the landscapes with numerous fruit trees, they had to learn mentally to monitor resources to be able to visit the right one again trees at the right time. Bats have been feeding for several months equipped with the GPS locators. This allowed to determine which trees were visiting and when. Bats have shown an extraordinary ability to create mental maps its environment by tracking the availability of food sources in space and time. In one experiment, scientists prevented bats leaving the colony for different periods, from one day to the week. We wanted to see if the bats could say that time has passed and adjust the behavior accordingly. We found that after 1 day of captivity bats returned to the trees visited the night before. However, after a whole week, older bats, based on earlier ones the experiences, they avoided trees that stopped fruiting at that time. In other words, they were able to estimate how long it's been since their last visit a concrete tree and knew which trees had been fruitful for a short time and not they were worth a visit. Young, inexperienced bats were unable to this is done, which indicates that this is an acquired skill that should be held. Lee Harden of Tel Aviv University said. Scientists also wanted to see if bats show future-oriented behaviors, whether they can plan ahead. This goal was observed by the roots of every bat. We found that bats they usually fly directly to a specific tree they know, sometimes distant 20 or 30 minutes. Being hungry, they fly faster when the tree is next, what he suggests they're planning where they're going. In addition, focused on the chosen goal, other trees pass, even good food sources visited only yesterday which indicates the ability to be delayed gratification. We also found out that the first bats leaving the colony choose fruit trees rich in sugar, while bats that leave it later, seek protein. Chen Sim, CO author of the publication, explained. All these findings suggest bats are planning preying before leaving the colony and know exactly where they are going and what kind of food they are looking for. Cognitive gap between people and animals are one of the most fascinating problems in science. Our study shows that nil-stats are capable of a fairly complex process decision-making involving 3 questions indicating abilities: cognitive, where to, setting the location of each tree, when is it. So it is knowledge of when a tree bears fruit. What is it? Knowledge of the topic food that provides sugar or protein. Once again we discover that the gap this one is not clear and that people are not as special as some people could think, Yovel said. A dog senses human stress and changes its response. The behavior. Dog's ability to sense stress in humans is known. But it turns out that 4-legged can infect a person's stress, which affects on the dog and his behavior. People can recognize when a person is stressed. The presence of such a person in the environment subconsciously influences emotions and choices made by other people. In new research, researchers from the University of Bristol found that dogs reacted similarly to stressed people. Results and description of the research appeared in the journal Scientific. The reports. These studies are the first, as the authors point out, which they check how the emotional state of dog caregivers can affect their pets. Dog owners know how sensitive their pets are to emotions. NER works show that even the smell of a stressed, unknown person influences the emotional state of the dog, perception of rewards and ability learning, said Nicola Rooney, lead author of the paper. A team of researchers invited 18 dog caregivers from with their pets. Animal caregivers were divided into two groups. One listened to relaxing music, and the other was solving mathematical tasks. Sweat samples were taken from all participants. At that time, the dogs underwent a little training. They were introduced to rooms where the bowl was. Animals were trained that when the bowl was placed in one specific place in the room, it contained the treat, but when she was in another, it was empty. Quadruples quickly learned the difference between locations. A bowl of mine. They were closer to a bowl with a treat. To the empty too they were zooming, but slower, as if only to see if it was empty like the last time. When it was already known that the dogs understood what the researchers were talking about, they began to place bowls in other places, except the two primary ones by location and how quickly the dog was approaching the new locations. A quick approach reflected optimism about presence food in bowls in new locations, a positive emotional state. From in turn, a slow approach indicated pessimism and negative emotions. The scholars then conducted a series of trials with smells. Each dog was exposed to the lack of smell or smells of sweat and samples breath from stressed or relaxed people. Fragrance samples stressed people came from a group of pet keepers who they were performing mathematical tasks. The latter from the group who listened to music. The results showed that dogs exposed to the smell of stress, slower they approached a bowl set outside of the two initial locations, however closer to the place where the empty bowl was originally located. The effect is not it was observed when animals were exposed to samples taken from the relaxed people. Researchers suggest that the smell of stress may have increased expectations of dogs that this new location does not contain any food, similarly like the nearby location, where the open bowl was initially empty. This one the pine-fistic reaction reflects a negative emotional state and may be a dog's way of saving energy and avoiding disappointment. Understanding how stress affects people. The well-being of dogs is an important factor in the case of dogs in shelters for animals and during the training of guide dogs, Rooney noted. The cooling power of sea ice is declining faster than the surface. The sea ice is getting less and less every year. Thus, the Earth's surface's ability to reflect sunlight reduces. It accelerates climate change. In a recent study, scientists determined that with the disappearance of sea ice and its decreased ability to reflect light, the Arctic has lost about 1 since 1984 of year cooling force. The Arctic and Antarctica are changing at a rapid pace. Ice. The sea is getting thinner every year and it is less and less. It follows from increase in temperature around the world. However, an increase in temperature in Reagan's polar is much faster than the global average. Sea ice performs functions similar to air conditioning in our houses, have cooling power of the entire planet. According to the university researchers of Michigan, his strength is no longer the same as it used to be. Satellite measurements, cloud cover and solar radiation reflected by sea ice in years 1980 to 2023 showed that the decrease in sea ice cooling capacity is more or less double the decrease in the average annual sea ice surface both in the Arctic and Antarctica. And the cooling power loss means greater impact of global warming. Results and description of the research appeared in the journal Geophysical Research Letters. Arctic has experienced the biggest declines in cooling power sea ice since 1980. Until recently, it seemed that the South Pole is more resistant to the changing climate. Sea ice cover on Antarctica remained relatively stable and its cooling power even increased. However, everything changed in 2016 when there was a violent loss. A lot of ice. Area larger than Texas melted on the continent's largest ice shelf, leading to significant loss sea ice and its cooling capacity. 7 more years. This is the weakest global effect of cooling sea ice since the early 1980s. With ice loss, the Earth's surface and increased rainfall create thinner, alpha and beta waves and appear slower, theta and the delta. In a new study, researchers found that sleep can be detected on the basis of patterns of neural activity lasting only milliseconds, i.e. 1,000 times less than a second, revealing a new way of research and understanding basic patterns of brain waves. They also showed that some areas of the brain they can temporarily exhibit sleep patterns during wakefulness and vice versa. These discoveries were made possible by artificial intelligence. Researchers have trained the neural network for the next few years to learn the function of sleep and how they affect behavior during sleep and wakefulness. A small part of the brain falls asleep when you are awake, or does that mean your behavior suddenly looks like you were sleeping? We began to notice that it is often the case, Schneider said. Observing the behavior of the mouse, the researchers noticed that when a region of the brain goes nap while the rest of the brain is awake, the mouse stops for a moment, as if it was suspended. In turn, reverse the situation, i.e. the wake-up of one area of the brain, while all the rest is sleeping, revealed in mice, the vibration of the body during sleep. Gaining a deeper understanding of sleep patterns and welding it can help scientists to better study neurodevelopmental and neurodegenerative diseases, which are associated with deregulation of sleep. Scientists want to continue their research. They also intend to use the brain organoids, bred in the laboratory, tiny versions of different organs that retain key anatomical features, the full-size organs. Mathematicians, altruism can be a survival mechanism. Humanity. Altruism is often seen as an attitude empirically impractical, which rarely brings measurable benefits. Meanwhile, simulations computer producing suggests it may be the mechanism of human survival, it informs the Krakow University of Technology. Jakob Schmiel, student of the second year of mathematics at the Polytechnic Krakowska and a member of the Student Scientific Club of PK Mathematicians, found mathematical proof of altruism can be seen as a mechanism the survival of the human species. The key to his considerations turned out to be the theory of games in mathematics and the prisoner's dilemma associated with it. The prisoner's dilemma is a known problem in game theory, based on a simple premise: each player can gain by betraying the opponent, but both they will lose if they betray at the same time. It is pictured like this: in a certain the two people are awarded the prize. Each of them can choose betrayal or cooperation, but they cannot communicate with each other. So they have to predict what the other person will do. If they both choose cooperation, each he will get 3,000 zlotys. If they both choose betrayal, 1,000 gold. If one person betrays and the other will collaborating, it's the one who chose the betrayal, will win 5,000, and the person that betrayed will get nothing, as she naively wanted to cooperate. Because in game theory, the player makes rational decisions, which are always as beneficial as possible. In this case, in theory, it always pays to betray the other person, because that is the case the behavior brings the greatest profits. As the PK informs, the prisoner's dilemma is not only the theoretical consideration, we are dealing with him in real life. An example is when two countries are fighting an arms race and they wonder whether to arm themselves or disarm. This game also occurs in various fields of science, such as economics, political science, or sociology. The first scientist who decided to explore the secrets altruism with the help of math and game theory was Robert Axelrod, American political scientist at the University of Michigan. Now, years later, his research Jakob Schmiel was interested. Based on them, he wrote a computer program repetitive dilemma of the prisoner between the wide populations playing different strategies. Examples of strategies, phenotypes, used in it. The solution is as follows. A traitor, always betrays. Sucker, always cooperates. Random, takes random decisions. Insulting, cooperates until the opponent again. The first will betray him, then he is offended once and for all. Wet for wet, starting with cooperation and then copying previous movement of the opponent. Forgiven, plays like in the wet for veto variant, but in 1 in 10 cases forgive, even if the opponent still continues to he cheats. Suspect, cooperates if your opponent cooperated so far, at least 50% of cases. Festarola, it starts from betrayal, then gives the opposite to the last move of the opponent. Cocker, stars cooperation, but if someone 3 times with rows cooperate or does not cooperate twice in a row, it betrays. Shitras, betrayal 2-3 times. The warrior, betrays 2 out of 5 times. Ready-made program checks what will happen to a random population playing the repeated dilemma of the prisoner over many generations, adding element promoting the strategy of those individuals who did better than the others. After many simulations, Jacob Schmiel noted that the population is usually dominated by individuals at some point distinguished by special features. Initially they are nice and start with cooperation, do not envy. In a single interaction always the opponents they win the same or more than themselves. They are honest, they do not betray as first, react to the betrayal of the opponent, and sometimes they just forgive. These features are very similar to altruism, to skill making good to others at the expense of your good, which may suggest why altruism occurs in nature. Finally, it is no wonder that if mathematical simulations show us the best strategy you can play in a betrayal situation or cooperation, after all, this best strategy is revealed in nature, he says. Jacob Schmiel. This program also makes self-updates under ambiguous of the preferences of these phenotypes that had the best results in the next generations of a population. This shows that in the overwhelming number cases in the game are won by individuals characterized by the mentioned previously, with features, and in the next generation, their number increases until total domination. According to the author of the program, mathematics shows us that to gain the most from the world around us, you have to behave like the winning phenotype, and therefore to be nice, unhealthy, honest, but at the same time do not let yourself be used and sometimes forgive. You just have to be good humans, Schmiel concludes. His program also shows that even in a situation where the population is starting from the position of overwhelming mistrust, and therefore frequent mutual betrayals, in the process of evolution, there are mutations that they change the player's point of view. Despite the harm suffered as a result of treacherous behaviors are more likely to decide for forgiveness. Alone like this individual could have done little, but a few such individuals could merge into groups and, for example, produce more goods than the competing groups that fought and betrayed each other. In the moment, when the representation of forgive people in the population reached a certain level, the society could have developed in a jumping way. Natural selection, he preferred the genes of such people. After many years, we observe that the result of evolution there are altruistic behaviors, Schmiel explains. Dr. Kamil Kuler from the Department of Applied Mathematics Krakowska, one of the guardians of the PK Mathematicians Scientific Circle, emphasizes that the project implemented by Schmiel is entirely based on meticulous statistical calculations. Of course, statistics have to do it is not always an accurate reflection of reality, but is the best way to tell her what we have. In this case, the figures confirm research hypotheses about altruism, he comments. The new method can pave the way to make the hardest element. Scientists have demonstrated a new way of manufacturing the super-heavy elements. Using this method, they were able to create Livermeter as the third heaviest element with atomic number 116. This is an achievement that opens the way to the synthesis of an element with an atomic number of 120. If it worked, it would be the heaviest element in the periodic table. In Lawrence Berkeley National Laboratory, LBNL, in the USA, the Livermeter was created the third heaviest element with a number atomic 116 in a way that can be used to create an element with an atomic number 120. Livermeter was created for the first time in 2000 by scholars from the United Institute for Nuclear Research in Dubna in Russia in cooperation with researchers from LBNL. The third heaviest element known in nature arose by breaking up the beam of atoms calcium-48 with a disk made of a Kairu. In new research, researchers from LBNL to the Lorvum produced was used by a beam of titanium-50 atoms and a plutonium disk. The team from Berkeley presented the results of the research at the conference Nuclear Structure 2024 in Lamont, USA. Description and results of the work have also been posted for publication in Physical Review Letters. You can now do with them read the AR14 preprint database. In LBNL was discovered so far, 16 of the 118 elements, and there are quite a chance to discover another, the heaviest of all. Super heavy elements are those that have a large number of protons in the test. This number is denoted with the letter Z and it is the so-called number the atomic. A few years ago, the periodic table was supplemented with 4 new elements. The heaviest of the new elements and the heaviest element in the system in general, the periodic, named Oganesson, has Z118. Named after the academic and explorer Yuri Oganessian. It was first obtained in 2002 as a result bombing of California 249 with calcium-48 atoms. Super heavy elements are very important from the scientific point of vision. They test the boundaries of nuclear theory, showing how many protons and neutrons it can coexist in the nucleus before it breaks down. And because the electrons in the inner shells of these atoms are so strongly attracted by abundance protons with the opposite charge, they spin at close speeds close to speed lights, distorting the shape of the nucleus. Synthesis of super heavy elements is simple, and at least it looks like it. Scientists launch lighter ions element into a thin target made of heavier element and hope that both nuclei will merge. The sum of the beam protons and shields are to be equal to the number protons in the atom you want to get. Both targets, missiles, and energies the bombings must be properly selected. The probability of happening expected nuclear reaction culminating in the formation of a testicle for a fraction of a second of the new composition is very small. Super heavy elements are defined as those that have Z above 104. Those with an atomic number between 105 and 118 were produced experimentally, but they are unstable and have very short half-life, therefore they are only academic and research. The latest super heavy elements with an atomic number from 114 up to 118 have been discovered using a calcium-48 beam. The number of protons and neutrons provides stability and a greater likelihood of connecting to the targets. However, this approach ended in oganesson, which has the atomic number 118, because the heaviest shields in which the bundles of ions go, create from California-249, which has 98 protons. Massive farms of 100 from him protons are falling apart too quickly to be used. Therefore, the physicists they began to use heavier but less stable elements. It comes out that a beam of 22 is needed to produce an element with an atomic number 120 protons, i.e. not calcium-48, which has 20 protons and titanium-50. Helmholtz Center for Heavy Ion Research, GSI, first started searching for the element 120 using a beam titanium that fired 4 trillion ions per second. However, the scientists they estimated that the experiment would have to last at least a year before it succeeds create a single atom 120. The search started did not bring any the result. GSI has suspended efforts since then, rebuilding the lab. In laboratories LBNL works the 88-inch cyclotron. In this accelerator, the scientists generate more powerful beams, which fires about 6 trillion ions per second. First of all, scientists heated the titanium-50 to 1800 degrees Celsius temperature, in which it began pair it up. With the help of microwaves, 12 of the 22 electrons were knocked out. Yes, of course prepared titanium was introduced into the accelerator and accelerated to about 11.9% of the speed of light. Then bombarded with a plutonium disk that rotated it will be 30 times per second to disperse the heat of collisions. Most of the ions go through an intact target. However, in rare direct impact combines titanium and plutonium. In the last experiments using titanium-50 beam and Platt-Ohlon detector blade in LBNL twice they recorded the 116 Lyrumer atoms, which fell apart within milliseconds flare 114 and then DIL 112. 2 Lyrumer atoms were obtained in 22 days of accelerator's work. This is much shorter than the researchers expected. According to them, it indicates that titanium and plutonium combine more easily. Scientists estimate that creating an element with a number the atomic 120 will take at least a few years after the start of work, if any it's gonna work. A platoon shield was used to produce a livermeter, but for the synthesis of the element with an atomic number of 120 will need a shield from the fillet Komodo varnishes have teeth covered with a layer of iron. The glories of Komodo varans are covered with iron coating which it helps to keep their serrated edges sharp as razor-like, the researchers determined. This adaptation certainly makes it easier for these animals to hunt and eat pregnants. Scientists suspect dinosaurs also had an iron layer on his teeth. Research conducted by researchers from King's College London shed new light on how Komodo whales keep their teeth resistant to Komodo consumption and sharp as razor. They also provide guidance on how dinosaurs like Tyrannosaurus rex killed and ate the victims. Results and description of the research appeared in the journal Nature Ecology and Evolution. Waran from Komodo, Varanus komodoensis, is the largest of the currently living lizards. It is sometimes called a Komodo dragon. This species is in danger of extinction. According to the International Union for Conservation of Nature, only about 3,500 dragons from Komodo remained freedom. Wherein from Komodo reaches a weight of up to 90 kilograms and length to 3 meters. These animals are strong, fast and able to cope. The victims are much bigger than themselves. But they will also be satisfied with a smaller prey. They eat almost every type of meat, from smaller reptiles and birds to deer or deer horses. But Komodo waranas have another impressive trait. There's razor-rough teeth, and they have 60 and exchange them all their lives. Covered they are in iron coating, which helps them in hunting. New tooth tests for these powerful predators revealed layers of iron along their serrated edges. The iron in the teeth is so much, that they're stained in orange. Many reptiles have some iron in their teeth, but for example crocodiles and other rentices have little of it, and often it's invisible. It's not just the reptiles that have made their teeth iron. Cryptochiton stellars, one of the types of mollusk, also adopted this strategy. In his own he uses teeth to scrape rocks to collect algae and bacteria. Iron in the teeth he lets him keep them for a long time. To understand the chemical and structural composition of vegan teeth of Vegan's Komodo, scientists searched museums in search of their skulls and teeth. They researched the teeth of Ganus, a 15-year-old Komodo dragon from the London Zoo that has been left dormant in February 2023 after being diagnosed with incurable degenerative inflammation of the joints. Advanced imaging and chemical analysis have shown that these animals concentrated iron along the cutting edges and the ends of the teeth. The protective layer makes the serrated edges of their teeth sharp and ready to use at any time. Iron also makes their teeth extremely hard and wear resistant. Moreover, the iron coating is also resistant to the acids. This can be good for lizards. These animals return hair, horns, and other indigestible foods, exposing your teeth to come into contact with its own digestive acids. These studies lead to new questions and directions of research how extinct species such as dinosaurs lived and fed. Komodo dragons have curved, serrated teeth with which they're tearing their prey, just like carnivorous dinosaurs. We want to use its similarity to learn more about how they could eat carnivorous dinosaurs and whether they used iron in their teeth in the same ways like Komodo dragons, said Aaron LeBlanc of King's College London, lead author of the study. Unfortunately, using the technology we have at our disposal at the moment, we can't tell if the fossilized teeth of dinosaurs had high levels of iron or not. We believe that the chemical changes occurring during the process of fossilization obscures how much iron was present at the beginning, he added. Researchers have found that larger carnivorous dinosaurs, such as tyrannosaurids, changed the structure of the enamel itself on the edges of their own teeth. While the Komodo dragons changed the chemical composition of their teeth, some dinosaurs have changed the structure of the enamel to keep the sharp edges the cutting. Thanks to further analysis of Varno teeth from Komodo, we can find other markers in the iron shell that do not change the phosphorylation. Such markers will allow you to determine whether dinosaurs also had teeth covered with iron, LeBlanc said. In search of a method of cleansing the Baltic Sea with weapons, it's chemical. According to estimates, at the bottom of the Baltic Sea lies approximate 40,000 tons of chemical weapons. These are all remnants of World War II. Various institutions and organizations have been trying to find a way to dispose of that deadly cargo. The new method will be tried by David Kramsky from the Wrocław University of Technology. The researcher will check whether the popular polymers used in 3D printing can be used to remove the arsenal at the bottom of the arsenal. David J. Kramsky from the Doctoral School of the University of Technology Wrocław received a grant of €30,000 from the Organization for the Department. Chemical Weapons, OPCW, ban, which will allow it to check whether popular polymers used in 3D printing after appropriate chemical modifications can be used to remove arsenic in the sea, the Baltic Sea. And there's a lot of it there. According to the Mayer Foundation, at the bottom of the Baltic Sea there is approximate 40,000 tons of chemical weapons, of which at least 15,000 tons are poisonous combat agents, BST, mainly sulfur iperite or arsenic, but also Clark I, Clark II, Adamsite, chloroacetophenone and tabun. It comes hard to estimate the amount of conventional weapons, aircraft bombs and mines. That's all this remnants of World War II. World War II chemical weapons depots. They are a significant source of arsenic in bottom waters and sediments. Arsenic, derived from decomposing chemical weapons, is a serious threat to the marine ecosystem and human health, emphasizes David Kramsky, who in the project wants to propose an innovative approach in the field of disposal the sunken war waste. The idea of making and testing the material to removal of toxins, printed on a 3D printer, fell during the research, which leads to his doctoral thesis. He deals with the subject removal of heavy metals on modified structures printed in 3D technology from polymer materials. My concept assumes obtaining an optimal structure, e.g. filter, 3D printed on a printer with commonly known and used polymers, e.g. PLA, ABS, PTGE, PC. Their advantages are that they are both it is easily accessible and cheap in obtaining, explains our PhD student. The next step in his project will be chemical modification of the printing surface so that it obtains sorption properties. Here it is the key will be the development by a young researcher from the PWR such method in order to the resulting structure had affinity for specific pollutants, that is, they catched, and in the case of his arsenic grant, appropriate material properties will result in intervention or permanent absorption of marine pollution, which I think is an attractive alternative to the existing methods, consisting in the extraction and destruction of sunken chemical weapons. David Kramsky explains. The Perseverance rover found an intriguing rock that it appears to contain traces of Martian life. The Perseverance rover movers of Mars. He discovered a rock that caught the attention of scientists. It contains features indicating chemical reactions that chemosynthetic microbes may have used to obtaining energy. Scientists remain cautious because to confirm the discovery further analysis is needed. On July 21, the Perseverance rover explored the northern edge Naretva Vallis, an ancient river valley about 400 meters wide, which has been carved in the ancient past by water falling into the crater Jezero. During his journey, he came across an intriguing rock. Analysis carried out by instruments on board the rover, it has shown that the rock has features that fit into the activity chemosynthetic microbes. Researchers are currently looking for other explanations, observed the features and emphasize that to confirm that these are indeed traces of ancient, microbiological life, further analysis will be needed. We designed a route for Perseverance so that we have reusability to reach areas that could potentially be a source interesting scientific samples, said Nicola Fox of NASA. The former riverbed in Naretva Vallis has paid off. We found something that we haven't seen before, she added. The discovered rock is about half a meter size. With its own shape resembles an arrowhead. NASA researchers refer to it as Shiava Falls, in honor of the highest waterfall in the Grand Canyon. Multiple times Sherlock, scanning habitable environments with Sherlock instrument, Raman and luminescence for organics and chemicals, mounted on rover, it has shown that it contains organic compounds. Although these types of particles carbon-based are considered the building block of life, they can also arise as a result of non-biological processes. It's the most enigmatic, complex, and potentially important the rock that Perseverance has so far studied, said Ken Farley of Caltech, involved in the Perseverance mission. On the one hand, we have the first convincing discovery of organic material, characteristic color spots indicating chemical reactions that microorganisms could be used as an energy source. We also have clear evidence that the water necessary for life once flowed because of that rock. On the other hand, we were not able to determine exactly how the rock formed and how the features visible on it were created, he added. Along the rocks run large, white veins of calcium sulfate. Between them there are strands of material, the reddish color of which suggests the presence of hematite, one of the minerals that gives Mars a characteristic rusty shade. When Perseverance looked closer to the red areas, discovered dozens of irregular, millimeters, cream-white spots, each surrounded by black material, similar to spots visible on the body. It's a leopard. Instrument PIXL, planetary instrument for X-ray lithochemistry. The Perseverance also has on board that he also determined that these black shells they contain both iron and phosphates. These spots are a great surprise, David said. Flannery, astrobiologist and member of the Perseverance team from Queensland University of Technology in Australia. On Earth, such features in rocks are often associated with the fossil record of microbes living under the surface, he added. These types of spots on sedimentary ground rocks can occur when chemical reactions involving hematite change the rock from red to white. These reactions can also release iron and phosphates, which can cause the formation of black envelopes. Reactions of this type can be a source of energy for microbes, which explains the relationship between such features and microbes in the environment. It's a land. Scientists believe that if we can find signs of life on Mars, it will be similar to Earth's life from its early days. It will be optimized for conditions with a low or zero oxygen concentration. The record the fossil life on Earth is difficult to study. It consists of layers fossil microbial mats placed between layers of rock, the sedimentary. In one of the scenarios considered by the team of scientists, this rock was initially a mule with an admixture of organic compounds, which on the space of time eventually cemented itself into the rock. Then the water flowing through the gaps in the rock, it provided mineral deposits, which they formed large, white calcium sulfate veins visible today and led to the formation of stains. Although both organic matter and spots are enjoyed great interest, these are not the only aspects of the rock that set the team scientific in perplexity. Visible white calcium sulfate veins are filled millimeter crystals of olives. This mineral may be linked to the rocks, which were formed in another part of the valley, probably as a result of crystallization, the magma. If so, the team has to answer another question, whether olivine and calcium sulfate may have appeared in the rock as a result of high temperatures and cause an abiotic chemical reaction that led to the formation of visible stains. We screened this rock with lasers and X-rays and imaged it literally at almost every possible angle. From Perseverance has nothing more to offer. In order to fully understand what really happened in the Martian Valley billions of years we'd like to take a sample from the rock back to Earth so you can to examine with the powerful instruments available in Earth laws, Farley said. It should be emphasized that the visible features of the rock are not unbending proof of ancient microbial life on Mars, at least for now. As NASA scientists pointed out, to confirm this, further research is needed, and it would be best if they were left carried out in Earth's laboratories, because Perseverance has exhausted the repertoire of research. This means that the newly discovered rock should be delivered to the Earth. The rover took samples of rock that will now await a spaceship that may one day arrive and take it back to Earth. Perhaps because the Mars sample return mission is facing considerable trouble with the financing. However, it may turn out that this discovery will make the problems of the mission they will disappear. Trees can absorb huge amounts of methane. It has long been known that trees preferably they affect the warming climate, removing carbon dioxide from the atmosphere. But they have another way to fight climate change. They can remove methane from the air, a strong greenhouse gas. This is happening for a case of microbes in the cortex. International team of scientists led by students University of Birmingham has shown that microbes living in tree bark remove from the atmosphere methane on a scale equal to or greater than the soil. The discovery highlights the importance of trees in mitigating climate change and supports efforts in their planting and forest protection as key strategies in global efforts for reduction of greenhouse gases. Results and description of the research appeared in the journal Nature. Methane has a much more impact on climate change than the carbon dioxide. It is 28 times more powerful greenhouse gas. His emissions have been rising sharply for nearly 2 decades, and researchers point out that it has contributed up to about a third of the observed climate warming since the time pre-industrial. This is a problem for the Earth's climate, because methane stops in atmospheric much more heat than equivalent carbon dioxide. But while CO may second EZ mainly keep methane in the atmosphere for hundreds of years, methane it has a much shorter lifespan, about 10 years. This means that the restriction methane emissions or streamlining processes that remove it from the atmosphere may be a faster way to mitigate increasing climate change. The sources of methane are both human activity, industry, agriculture, landfills, as well as natural processes taking place in the swamps and the wetlands. Most of the methane emitted is removed by processes that occurs in the atmosphere or in the soil. These are full of bacteria that absorb gas and use it to obtain energy. Until now, the soil was believed he's the only terrestrial methane absorber, but scientists now show that trees can be just as important and perhaps even more important. During photosynthesis, tree leaves absorb the beneath coal and store it as biomass in trunks and branches. New research shows that there is another way that trees absorb greenhouse gases. This way forests can provide even more climate benefits than it was previously thought. It turns out that the bark of trees in forests around the world it absorbs methane. Scientists in new analyses looked at the bark of trees, previously overlooked in studies on limiting change climate. The main way we are considering the contribution of trees to the environment is the absorption of carbon dioxide through photosynthesis and storage. However, our research shows an extraordinary new way that trees testify an important climate service. Global Methane Commitment announced in 2021 at 26 Colombian pesos Climate Summit aims to reduce gas emissions 30% by the end of the decade. Our work suggests that planting more trees and reducing deforestation must be important elements of each approach this is done, said Professor Vincent Gauci of the University of Birmingham, lead author of the research. Wetlands are the main natural source of methane. The trees in swamps and floodplains can emit methane from their lower parts, but the exchange of methane in trees growing on well-permeable soils and not independent most of the world's forests have not been so far it was analyzed. Scientists have studied forest tropical, boreal, and moderate zones, and specifically from forests tropical in the Amazon and Panama, deciduous trees in Waltham Woods in Oxfordshire in the UK and coniferous forests in Sweden. The absorption methane was the strongest in the tropical forests, probably because microbes develop better in the warm and humid conditions prevailing there. On average, the newly discovered methane uptake adds about 10% to the benefits climate provided by tropical forest trees and zones the moderates. Researchers used simple plastic wrapping chambers around the trees. They were connected to them by methane detectors. It turned out that some trees they actually emit a small amount of methane from the base of the trunk. But the higher trees were putting on their chambers, the greater the absorption of this gas. Scientists have also been able to determine the total surface area of the bark trees in the world. Thanks to laser scanning, they mapped the bark all the way to the thinnest twigs. They found that if they were to spread the bark from all the trees to the world would cover the entire surface of the Earth's land. This is a huge area gas exchange between the cortex and the atmosphere, but this mechanism is still poor heat is known. Preliminary estimates by researchers suggest that trees depreciate from approximately 25 to 50 million tons of atmospheric methane per year, most of them they absorb the tropical forests. This is a similar result to another methane absorber on land soil. But unlike soil, the surface of which is not it changes, the forests shrink and expand through deforestation and reforestation. These changes can affect atmospheric methane. It is clear that decarbonization of the global economy and the energy system is a key way to solve the problem of change the climate. But the newly discovered tree bark ability to absorb methane offers a new way to fight this problem. Planting trees, selecting species that are particularly good at removing methane from the atmosphere or modifying tree bark microbial communities can complement our arsenal in combat with global warming. Study authors emphasize that it is necessary to protect existing natural forests and halt further deforestation. Studies on twins have shown a beneficial effect of diet vegan for aging process. In new studies involving 21 couples twins, one of them followed a vegan diet, while the other did not. It has in no way limited the food we eat. The experiment was 8 weeks and demonstrate could short-term processes aging and lower the biological age. Every person has a certain chronological age, which counts from the date of birth. There is also a biological age that is set for the basis of aging of the body's functions. This age reflects the health of striatal tissues and tissues of a person, and the influence on it can be affected by diseases, style life, environmental factors, and many others. In new work, scientists looked at the influence of the transition for a vegan diet. They invited 21 pairs of identical twins. The trial consisted of 77% of women, and participants were 40 years old on average. Scholars they wanted to see what changes are happening when one of them is following a diet vegan, and the diet of the other includes everything, including meat, eggs and dairy every day. The experiment lasted 2 months. For the first 4 weeks, the participants ate meals specially prepared for them, and by the next few weeks prepared them themselves after taking short courses the cooking. Research suggests that the use of a vegan diet by a few months can slow down aging process and lower biological age. The authors emphasize, however, that the results that appeared in the journal BMC Medicine should be treated with reserve, partly due to restrictions in the way the test is conducted. Study authors looked at changes in methylation levels DNA from the twins. DNA methylation process leads to changes in the activity of some genes without modifying the original DNA sequence. To determine the state of health, human cells and tissues are using levels of DNA methylation. These patterns change throughout life and progress with age. Biological person who may be greater or less than age measured in the years. In general, increased DNA methylation is considered a marker of progressive aging processes. DNA methylation can be analyzed by taking blood samples. That's why they were downloaded from study participants at the beginning, in the 4th and 8th week of the study. They were then compared to see if and how to change the diet it affected the subjects. Scientists used the level of DNA methylation to infer the biological age of the participants. Interestingly, researchers have observed a decrease in age estimates biological in participants who followed a vegan diet, but not in those who ate everything. In people who have switched to a vegan diet, they also noticed reduction of aging markers for the heart, liver and inflammatory systems and the metabolic. However, the authors point out that it is not clear in which the extent of the observed differences between participants can be attributed to the diet. Participants on the vegan diet lost an average of 2 kilograms more than their omnivorous siblings, because they took in less calories. Researchers indicate that these differences in weight loss may have contributed to observed differences in epigenetic age between the two groups. In the other one, studies have shown that calorie restriction slows aging in mice and the monkeys. Although it is interesting to observe the reduction of biological age after such a short time, the length of the test also means that it has not included long-term effects of changes in diet. If the vegan diet is not supplemented with vitamin B12, the vitamin B12 deficiency develops, which causes chronic and often insidious damage to the nervous system, said Tom Sanders, Emeritus Professor Nutrition and Dietetics from King's College London. Long-term research observation vegans also show adverse effects on density bone, which is probably due to a very low calcium intake in the protein. Although observational studies indicate that vegan diets may have beneficial health effects in middle ages, such as lower risk of cardiovascular disease and diabetes type 2, this does not apply to older vegans who seem to be more exposed to loss of muscle mass, low bone density and disorders neurological, which has a significant impact on the quality of life, he added. Authors of the publication indicate that further research is needed to establish a link between the composition of the diet, weight, and aging processes. Bacteria that dissolve certain types have been discovered. The cancers. Commonly occurring type of bacteria in the mouth, it can be used to fight cancer. Fusobacteria, because we are talking about them, they appear to have the ability to dissolve head and neck cancers, according to new research. These works have the potential to create new, effective methods of treatment. The type of cancer. Fusobacters are a type of bacteria commonly found in the mouth. Until now, they were associated with accelerating development the colorectal cancers. In other analyses, it is established that they may also contribute to the development of endometriosis. But in new research, scientists from the Network Guy's and St. Thomas's and King's College London hospitals have shown that fusobacters are very good with head and neck cancers. In people with these cancers who have been diagnosed with the presence of fusobacteres in their tumors, the results of treatment are much better. Scientists are studying now the exact mechanisms behind this connection. Results and description of the research appeared in the journal Cancer Communications. Head and neck cancers are a group of cancers for which it includes JAMA cancer, oral, tongue, throat, larynx, glands, cavities, nasal sinuses, ear and thyroid. They are the sixth the most common cancer in the world, and among their group the most common is larynx cancer. In Poland, they are diagnosed every year in 11,000 people. This group of cancers is more common to men and it depends on environmental factors and lifestyle. The biggest risk the development of head and neck cancers affects people who smoke cigarettes and they consume alcohol. Survival rates depend on what stage the disease was detected. In recent years, there has been no significant progress in their treatment. Till it now. Dr. Miguel Reis Ferreira and his co-workers, he found that patients with head and neck cancer who were found to have higher levels fusobacters, had better prognosis than patients with lower levels the bacteria. These observations have intensified the research on Fusobacters. Dr. Anjali Chander from King's College London begins breeding cellular head and neck cancers and found that the number of living cells cancer in its breeding decreased by 70 to 99% after infection the Fusobacteria. We found that when these bacteria are present in cancer head and neck, they have much better treatment outcomes. Another thing we've discovered is that in cell cultures these bacteria they are able to kill cancer, Ferreira said, adding that the bacteria contribute to better treatment results based on what they do inside the tumor. They probably destroy cancer by releasing toxins that they reduce their service life. Currently, we are looking for this mechanism. It should be the topic of the new publication in the near future, he added. Our research reveals that these bacteria play more a complex role in cancer than previously thought. They basically dissolve cells of head and neck cancer. However, this discovery should be balanced by their known role in the development of cancers, such as those in the gut, he stressed Ferreira. Analysis of data of 155 patients with head and neck cancer, of which the information was sent to the Cancer Genome Atlas database showed that people with cancer Fusobacteria had a better chance for survival compared to those who weren't there. The presence of these bacteria in head and neck cancers, a 65% reduction in the risk of death was associated with a 65% reduction in compared with patients whose cancers did not contain Fusobacteria. These studies can help to deepen your knowledge of bacteria and cancer interactions. They can also lead to development new, effective treatments for this type of cancer. We want better understand how we can translate the newly acquired knowledge into improvements in the treatment of patients with cancers of the head and neck and other cancers, Ferreira said. Chinese scientists discover water in samples brought from the moon. A team of Chinese scientists has discovered closed-in minerals found in samples of lunar soil delivered to Earth by Chang'e 5 mission in 2020. Researchers found that water on the moon can survive even in areas illuminated by the sun as the hydrated salts. The Chang'e 5 mission lasted just over 3 weeks. Within the framework the mission lander drilled into the lunar ground to a depth of about 2 meters, where he took samples of matter. The material was also taken from the surface of the moon. The samples were then delivered to Earth. It was the first samples from the moon brought to Earth since the 1970s. The 20th century. The material was downloaded near Mons volcanic formation Rempure, located on the northern edge of Oceanus Procellarum, Latin. The Ocean of Birds. Mons Rempure is a volcanic formation with a diameter of about 70 kilometers erecting it's over a kilometer high. Scientists believe that about 1.3 billion more years ago, this volcano may have been active. Ocean storms, on the other hand, is large, covered frozen basalt plain, visible from the Earth as a dark structure on the west the edge of the visible side of the Moon. Previously this region of our natural the satellite was not visited. Analysis of the material collected from the Moon showed the presence of water. The fact that there is water on our natural satellite, we have known for at least a decade. Moreover, scientists found traces of water in recent analyses of samples brought to Earth in the 1960s and 1970s. XX of the age. However, the samples collected by the Chang'e 5 mission come from significantly higher latitude, which gives new clues, what form it receives water on silver globe surface and creates new perspectives for use in exploration of the Moon's resources. Results and description of the research appeared in the journal Nature Astronomy. Scientists in the publication describe the discovery in samples lunar carnallite regolith, hydrated double chloride potassium and magnesium. They also found a hydrated mineral whose structure and the composition resembles novogribolnauite, a mineral found near volcanoes on Earth that arises as a result of the reaction of a hot basalt with water-rich WAD gases the volcanic. This mineral may have been formed as a result of volcanic eruptions on Moon, said Jin Shifeng, a member of a research team from China Academy of Sciences. This indicates that volcanic gases on the Moon contain a significant amount of water, he added. These findings revealed a potential form in which the water molecules WADI can appear on the surface of the Moon, i.e. hydrated salts. Such hydrates are very stable. They can occur even in areas illuminated by the sunday. Due to high temperatures and no atmosphere the Moon, the existence of liquid water is not possible, So before not it was clear how the water molecules could exist there, Jin said. Remotely controlled mice. South Korean scientists have presented the technology through which to manipulate specific areas of the brain with fields the magnetic. In experiments, they forced mice to eat or socialize. The technology this could potentially reveal the secrets of advanced brain functions, such as knowledge, emotions, and motivation. Researchers at Yonsei University in South Korea have developed a new method of controlling the brain with magnetic fields. The nanomind technology, magnetogenetic interface for neurodynamics, enables wireless, remote and precise modulation of specific areas of the brain with magnetic fields. In the experiments on mice, scientists have shown that it has potential for remote control of emotions, social behavior, and appetite. Description and results of research appeared in the journal Nature Nanotechnology. The human brain contains over 100 billion neurons connected in a complex network. Neural circuit control is crucial for understanding higher brain functions, such as cognition, emotions, and social behaviors, and also to identify the causes of various disorders. The new technology controlling brain function is also important for interface development brain-computer, BCI, such as those developed by the company associated with Elon Musk Neuralink, whose purpose is to enable device control external only by means of thought. Attempts to control animal behavior are nothing new. However, in previous work, electrodes were usually used attached to the head of the animal connected to the external system. Such as the one the approach requires invasive operation and does not allow you to move freely the test objects. South Korean researchers have developed a different system. In the owls the work used the magnetic fields, which allows wireless and remote control specific areas of the brain to modulate its complex functions. Although magnetic fields have long been used in medical imaging for their safety and ability to penetrate biological tissues, precise control of neural circuits with magnetic fields it was a serious challenge for scientists. This advanced technology uses magnetic fields and magnetized nanoparticles for selective activation of target circuits the brain. Scientists have genetically modified mice to make those they developed more magnetically sensitive ion channels that act as the gates in their neurons or cells of the nervous system, allowing for sure molds enter at certain moments and at a certain speed. Selective expression of nanomagnetoreceptors in specific types of neurons and activating them using magnetic fields at precise moments, spatial temporary control of neural activity. In the experiments, the researchers showed that their method works. It helped with the maternity behavior of the mice. Activated a specific A group of neurons in non-mother myelin females, which significantly increased them care behavior towards the young. They reacted completely differently to the calls the young, approached them faster and spent more time with them. They also used their technology to regulate behavior nutritional genetically modified rodents. Activation of the neurons caused the mice to eat only half the portion. In another the researchers, in turn, caused an increase in appetite. In another one more the experiment inspired the mouse to a friendly attitude to the second mouse. Experiment results show that nanomind technology could selectively activate desired brain circuits to modulate higher functions brain. This is the world's first technology to control specific areas brain with magnetic fields, says Jinwoo Cheon, co-author of the paper. We expect to be widely used in research to understand function brain, advanced artificial neural networks, bidirectional technologies BCI and new methods of treating neurological disorders, he adds. Moderate consumption of alcohol, however, does not prolong life and not good for health. Excessive alcohol consumption is well documented a danger to health. However, occasionally you can hear about the beneficial effect of the consumption of alcohol. Scientists in a new meta-analysis have questioned these findings, saying it's the result of bad research. Research suggesting that alcohol may be beneficial for health and even extend life if consumed in moderate quantities, can be based on faulty research methods. This is according to Journal of Studies on Alcohol and Drugs meta-analysis by researchers from the University of Victoria in Canada. Among many of us there is a belief that moderately drinking alcohol is associated with the supposed health benefits. Many people say that drinks a glass of wine because it works well for the heart, or a small beer, because it brings benefits of the kidneys. But from a recent publication, These beliefs are the result of poorly conducted studies whose authors have exaggerated the benefits health from moderate alcohol consumption. Canadian scientists have analyzed 107 studies alcohol consumption, drinking patterns, and how long the drinkers live. In the research, a total of 4,838,825 people participated. Scientists have noticed that in the majority cases in the study compared drinkers with those who stopped, they were drinking or consuming very little alcohol, not considering that some have limited or stopped drinking because of their ill health. This means among abstinences and occasional a significant number of patients drink is a significant number of patients, which reduces the average health status the study group and makes people drinking little or moderate they perform better compared to them. Analysis of more than 100 studies actually clue to pull out conclusion that moderate alcohol consumption, 1 drink per week to 2 on average, 14% reduction in the risk of death compared to with the abstinence. However, when the authors of meta-analysis delved into the work, the picture it has changed. Canadian researchers have divided the study by quality. According to the opinion, those of high quality are those in which the average age of the cohort was 55 years or less, in which the observation lasted more than 55 years, and in which they were or occasionally drinkers were excluded from the reference group. This allowed to so studies involving persons abstaining from alcohol, but with a history of drinking. It turned out that in the highest quality research was not evidence that moderately drinkers lived longer. Research of the lower quality, those involving older participants who did not distinguish between ex-drinkers and abstinence from birth, were the ones that combined alcohol consumption with a longer life. If you look at the weakest research, it's there you will see health benefits, said Dr. Tim Stockwell, lead author the publication. Estimates for the health benefits of drinking alcohol, they have been exaggerated and his damages underestimated in most of the previous ones. Research. The vast majority of them compare drinking with a group of people who are now they abstain from alcohol or drink very little. We know people give up or limit drinking when the sick and weaker become older. The most viscous studies included those in the control group who stopped or reduced drinking for health reasons, so people healthy enough strove to continue drinking. They seem even healthier, Stockwell added. The authors of the meta-analysis emphasize that there is no safe for the health of the dose the alcohol. Surprising discovery in the 16th century laboratory. The astronomer. Danish scientists conducted a glass analysis in ceramic vasodilators found 35 years ago in the place where once there was a laboratory of Tycho Brahe, an astronomer living in the 16th. The age. Brahe, like many scientists of the time, was also involved in alchemy. Research on the remains from his studio brought a surprise. They showed traces the element, which was only officially discovered only 2 centuries later. New research gives some tips on what, besides astronomy, he was involved in. Tycho Brahe. Brahe is remembered today as the greatest astronomer of the eras before the telescope was invented. Unprecedented precision he achieved in measurements of the planet's location proved necessary to explain their movement by Johannes Kepler, who was his assistant. But Brahe, outside of astronomy, he was also interested in alchemy, as many scholars of his time. It is not he was looking for a way to turn base metal into gold, but tried to use they are for treating diseases such as plague or syphilis. In his workshop he used mercury, gold, various herbs, or minerals. However, little is known about the Danish work at the moment. The scientist. Alchemists were known for their mystery and did not share with the other knowledge gained. Brahe was no exception. Just a few of his alchemicals the recipes survived, and its work in the basement of the residence the island of Venus, which belongs to Sweden today, was destroyed after his death. Among the preserved recipes of Brahe are found, among others, Ateria, popular in those time the drug, which contained about 60 ingredients and which was recreated by scientists from Wrocław. In 1988 to 1990, the Brahe mansion was conducted in the Brahe residence excavations. Dozens of ceramics and glass were found. He believes these pieces are from Brahe's alchemical studio. 5 of these shrapnel, 4 glass and 1 ceramic, undergone in recent tests detailed chemical analysis to determine what elements they had with contact original glass and ceramic dishes. All except 1 glass shrapnel showed elevated concentrations relative to background levels: nickel, copper, zinc, tin, antimony, tungsten, gold, mercury, and lead. It is up to Phi. It indicates that each of these elements was used in experiments led by Bra. Most of the discovered elements are not surprising, especially when it comes to an alchemist laboratory. The most intriguing discovery there is the presence of tungsten. Wolfram is very mysterious, said the Professor Carl Lund Rasmussen from the University of Southern Denmark, which he conducted chemical analysis. Wolfram in those days was not even described, he added. Wolfram was first described and produced in pure figure in 1781, more than 180 years after Bra's death, by Swedish chemist Carla Wilhelm Scheele. Wolfram occurs naturally in some minerals, and perhaps this element went to Bra lab for the mediation of one of these minerals. In the lab, the mineral may have been subjected to a certain treatment that separated the tungsten, without Bra's knowledge. However, there is another possibility. In the first half the 16th century, German mineralog Georgius Agricola reported that tin ore from Saxony contains a mysterious ingredient he named wolfram, in German wolf foam, which made it difficult to melt them. Maybe Tycho Brahe heard about it and knew about existence. It's a tungsten. But it's not something we know or we can say on the basis analysis by me. This is just a possible theoretical explanation, why we find tungsten in the samples, Rasmussen said. Research from the University of Southern Denmark and the Danish National Museum in Copenhagen has been published in a magazine, Heritage Science. The Great Red Spot on Jupiter is shrinking. All of it, by changing the diet. The Great Red Spot is the most characteristic A feature of Jupiter. It's a storm whose diameter is larger than the diameter of the Earth. In recent years, astronomers have noticed that the Great Red Spot is shrinking. New research suggests that the reason for this is a change in diet. The Great Red Spot in Jupiter is the biggest storm in the solar system. It has been observed for centuries. It can be seen on Jupiter's southern hemisphere as a giant oval of reddish clouds that rotate in counterclockwise at wind speeds up to 680 km per hour. The size of this structure is changing. At the end of the 19th century, the Great Red Spot was 39,000 kilometers wide. Now it has 14,000 miles of width. Earth diameter is 12,742 kilometers, which means that she could fit in. But this structure is still declining, and a new study could help explain why. Many people have watched the Great Red Spot by the last 200 years, and it was as fascinated as I am, Caleb said. Keaveney of Yale University, lead author of the new study, which appeared on the journal Icarus. Many of these people were not professional astronomers, they were simply passionate and curious, he added. This curiosity is fueled by many mysteries that they surround this structure. Despite the fact that it has been studied many times, astronomers don't know exactly when it was created, why it was created, or even why it has a red-orange color. It seems that at least one of the mysteries about the Great Red Spot have been solved. Scientists believe the Great Red Spot draws energy from other smaller storms that connect with it. If these smaller storms, which the Great Red Spot could absorb, is short, the structure is not able to keep your size. Scientists conducted a series of 3D simulations red spots using the atmospheric model for planetary applications developed by Timothy Dowling of the University of Louisville in the 90s. The 20th century. Some of them simulated the interactions between the big red spot and smaller storms of different frequencies and intensity. In the other simulations have been omitted small storms. Comparison of the simulation showed that the presence of other storms reinforced the Great Red Spot, causing it the magnification. Thanks to simulations, we discovered that by feeding the Great Red diet stained with smaller storms, as it happens on Jupiter, we can to modulate its size, Keaveney said. However, in the last decades there has been significant decline in the number of small storms, and with them the Great has decreased the red spot. Scientists based their modeling partly on long-lasting high-pressure systems observed in the Earth's atmosphere. These systems, known as heat domes, occur regularly, circulating through medium-width Earth's geographical and play a big role in extreme weather events, such as heat waves and droughts. The life expectancy of these systems has been linked to interactions with smaller, transient weather mechanisms including vortices, high pressure and anticyclones. Our study has implications for weather phenomena on Earth, Keaveney said. It has been shown that interactions with nearby systems weather support and amplify the heat domes, which justified are the idea that similar interactions in Jupiter could sustain the Great Red Spot. Confirming this hypothesis, we provide additional support for understanding the heat domes on Earth, he added. Keaveney admits that additional modeling will allow researchers excel new discoveries and perhaps shed new light on the formation process. It's the Great Red Spot. A new era in HIV prevention? Injection twice a year, it provides 100% protection. Research involving more than 5,000 young women, they showed that the injection twice a year of an antiviral medicine called lenacapavir protects them 100% against HIV infection. This measure has great potential and can change the prevention of HIV infection. Results of a recent Purpose 1 study were better than whoever was expecting. Antiviral agent called lenacapavir, having to combat human immunodeficiency virus, HIV, infection has been approved last year by the US FDA and EU regulatory authorities European for Pre-Exposure Prevention of HIV Infection, PrEP. Studies on the effectiveness of the drug were carried out in South Africa, Uganda, and the United States. 5,338 girls and women aged 16-26 took part. Years. None of them were infected with HIV. The participants were divided into 3 groups. The first was given lenacapavir injections every 6 months. They were women and girls from South Africa and Uganda. The group consisted of 2,134 people. The other 2 groups, they received daily emtricitabine and tenofovir alafenamide tablet. Approved in many countries as PrEP, or a newer version of the tablet that has fewer side effects, or emtricitabine with tenofovir disoproxil. In the group using lenacapavir injections twice a year, no HIV infection has been reported. Among the other 2 groups were 55 new HIV infections. However, in these 2 groups, as the blood tests showed, most women took 3 or less pills per week instead of 1 a day. People taking the least tablets were most likely to an HIV infection. Scientists indicate that patterns consisting of 1 tablets daily did not reduce the risk in young women in high-ranking countries' HIV load, because factors such as stigma or dependence from other members of the community stop many women from receiving tablets as directed. From this point of view, less frequent administration of the drug, it can have a greater effect. The results of the study were published in the journal The New England Journal of Medicine. They were also discussed at the recently completed 25. The International Conference on AIDS, which is being done in Munich. Lenacapavir is injected into the subcutaneous tissue, usually in the stomach. It is the first drug of this type that works on HIV capsid protein, the shell of the virus, protecting the genetic material and enzymes needed for the replication. Capsidox plays a key role in many stages of the life cycle of the virus, and it is a good target for medicines. HIV surrounds its genetic material with a protein envelope to protect the virus from host defense, designed to inhibit the virus the infection. Developed by the biopharmaceutical company Gilead Sciences, lenacapavir, it was designed to deprive HIV of a protective envelope. This measure is strong, safe, and working for a long time. Joining the capsid the surrounding HIV and interrupts the life cycle of the virus. Makes HIV capsid very brittle, leading to its rupture before the virus disappears for good in the host cells. When a virus loses a protective shell, it becomes easy target for the host's defense. So far, scientists have not observed any signs of resistance development HIV for the drug, but, as they emphasize, it is probably a matter of time. For example, when a person stops taking the agent regularly, he maintains in the body for weeks in concentrations low enough to allow the virus survival and adaptation. In the United States and in 6 other countries it was a Purpose 2 study in which men participated. Data from the trials are expected at the end of this year or early next year. Consuming too much sugar may accelerate aging processes in women. Using a diet containing a large amount of sugar can have an adverse effect on biological age, according to studies conducted with the participation of women. Age is usually defined as the number of years lived, but it can also be determined in other ways, more precisely, on the basis of pace of the aging process. In recent years, scientists have developed ways to measure biological age by tracking chemical changes in the DNA. They progress with age. but appear at different times people. These so-called epigenetic clocks indicate the age of the body more accurately. The epigenetic clock is based on the study of the methylation process, the DNA. In this process, methyl groups attach DNA and lead to changes in activity of some genes without modifying the original DNA sequence. Patterns DNA methylations change throughout life and progress with age. Biological person who may be greater or less than age measured in the years. Scientists can determine someone's biological age, measuring the chemical changes in our DNA that occur over time. New study of 342 women in the middle age has shown that consuming foods with plenty of added sugar for the biological age. Researchers studying samples from women admitted that cells of those participants whose diet contained a large dose of sugar, they look older than their age. These findings, described in the journal JAMA Network Open, they can help explain why some people seem to age faster or more slowly than others who have lived the same age. Sugar consumption can be an important but overlooked factor. Every gram of added sugar is associated with an increase in age epigenetic, even following a healthy diet. In turn, a diet rich in ingredients nougat, especially one that does not contain a large amount of sugar, can slow down the biological rate of the body's aging. Studies have shown that women in middle age, which ate more foods rich in vitamins, minerals, and antioxidants had younger-looking cells. Other of healthy diets they were linked to the slowed aging process, and the addition of sugar was spent to suppress this effect. Researchers evaluated cell age based on methyl groups on the surface of DNA. Larger amounts of added sugars in the diet seem to accelerate aging processes, even if women have followed a healthy diet. Added sugars refer to sugars added to food during the production. It is about sugar-sweetened drinks, cucumber confectionery, in contrast to naturally occurring sugars in fruits and vegetables. We knew that high levels of sugars added in the diet is associated with metabolic health and various diseases, probably more than any other factor dietary, said Alyssa Eppel from the University of California, San Francisco, co-author of the study. Now we know that accelerated aging epigenetic is at the heart of this relationship, and it is probably one of many how excessive sugar consumption reduces longevity, he added. Researchers analyzed the 342 diet in examination of the women. The analyses concerned products consumed by women within the course of 3 not consecutive days. Then they tried to match habits nutrition women for various diets, such as the Mediterranean diet, rich in plants, whole grains, and unsaturated fats, and low in red meat, saturated fats, and sugars. For research, they also developed a measure nutrient intake they called the Epigenetic Nutrient Index. This measure includes nutrients linked to antioxidant processes and anti-inflammatory in the body, as well as related to the maintenance and repair of DNA. For example, vitamin A, C, B12 and E, as well as folic acid and magnesium. In addition to the evaluation of the research participants' diets, the team evaluated also how much added sugars were consumed by women. Here the results ranged from 2.7 up to 316 grams of added sugar to every 1 day. Milk chocolate table contains about 25 grams of added sugar, and a can of collars about 35 grams. Finally, scientists calculated the epigenetic age of the women. These data showed the links between diet and aging the cells, but captured only part of the image. Research confirms the thesis that consuming nutritious foods low added sugars can improve health and influence of longevity. However, more research is needed to assess in how following these recommendations may affect aging epigenetic in the long term. It is known that diet, style life, genetics, and diseases affect the speed of ticking the epigenetic clock a person, but this is one of the first studies to show what role it plays sugar in aging process of the body. Given that epigenetic patterns seem reversible, eliminating 10 grams of added sugar per day, it may be equivalent to the withdrawal of the biological clock by 2.4 months if it will last for a long time, said Barbara Laraya from the University of California, Berkeley, co-author of the publication. Focusing on products with high content of key nutrients and low the content of added sugar can be a new way to motivate people to eat healthy longevity, she added. Scientists have discovered a new type of wood that may be highly efficient in cold storage. The tulips have a completely different wood structure than other species of trees. Research from Jagiellonian University and University of Science Cambridge. This wood seems to be particularly efficient in storing coal. This discovery could open up new opportunities for improving carbon sequestration in the new planted forests. Tulip oak trees are happy in gardens because of its own beauty. They are related to magnolias and can reach a height of over 60 memoirs. Naturally, they can be found in North America, Liriodendron tulipifer, and in China and Vietnam, Liriodendron chinense. Bini, the first species, was used by the Native American population northern to make canoe. Indians also made a drink from roots of the curative. Currently, tulips are made from furniture. In the new research, scientists have discovered that these trees have a unique type of wood, which does not fit or wood of hardwoods, nor coniferous trees. Results and description of the research appeared in the journal New the Phytologist. Researchers from the Jagiellonian University and University of Cambridge conducted a microscopic wood structure test from some of the most famous trees and shrubs in the world. In total, they subjected 33 analyses the species. Research was meant to know how wood structure it evolved in coniferous and deciduous wood. Scientists also checked the structure macrofibrillary, fibrous structures with a diameter of 10 to 40 nm forming wood. Using a low temperature scanning electron microscope, they found that 2 living species of tulips have much larger macrophylicals than other studied species. We have shown that the tulips have intermediate macrofibril structure, which is significantly different from the structure of hard. These species separated from the magnolia trees about 30 to 50 million centuries ago, which coincided with a sharp decline in atmospheric CO2. Maybe it has helped explain why tulip trees are so effective in storage coal, said Dr. Jan Lizakowski from the Plant Biotechnology Department of the Faculty Biochemistry, Biophysics, and Biotechnology of the Jagiellonian University. It is known that both tulip species are extremely efficient in binding coal, and their enlarged macrofiber structure can be an adaptation, which helps them more easily capture and store more coal when the availability of atmospheric carbon is limited. Tulips can be useful in coal capturing plantations. Some of the Asian countries eastern is already using the plantations of these trees to effective bonding of coal, and now we think it may be related to their new discovered wood structure, he pointed out. Likakovsky, a researcher from the Jagiellonian, admitted that despite the importance of the trees, not much we know how the wood structure evolves and adapts to the environment external. The main building blocks of wood are cell walls, secondary. The architecture of these cell walls gives wood density and the strength we rely on in the constructions. Cellular walls are also the largest carbon repository in the biosphere, which makes understanding their diversity is even more important to develop our carbon capture programs to mitigate change climate, emphasized Likakovsky. Studying wood ultrastructure is crucial for various applications, including woodworking, sciences, materials, and understanding of the ecological and evolutionary aspects of trees. Understanding the biology behind tree growth is also valuable information. Calculation of carbon capture. Great news for bald people. This one, the success of scientists can herald a breakthrough. Biotechnologists from Japan in laboratory breeding managed to get fully functioning mouse hair follicles. It's definitely good a message for people struggling with baldness. The achievements of scientists can open the way to regenerative baldness therapies, as well as drug testing and study of hair growth disorders. Hair follicles have bred a team of researchers from Yokohama National University in Japan. Formation of hair follicles during the development of the body. It has been studied for several decades, but has not been able to recreate it into the artificial conditions. Recently, however, there have been further successes in the creation of so-called organoids, miniature models of different organs. Such as the one structures are used, among others, for the study of diseases, potential therapies, and development the organs. Organoids have become a promising tool for understanding the mechanism of creating hair follicles in vitro, he explains. Professor Tatsudo Kageyama, author of the paper published in the magazine Science Advances. Researchers used 2 types of mouse, parent embryonic cells placed on a special basis controlling them. It's an increase. With almost 100% efficiency, hair follicles developed from them with the stems of hair. Scientists have added to them a substance that stimulates development of pigment-forming cells, melanocytes, which gave the glands of hair the color. In the next step they implanted grown bellows into the skin of the mice, obtaining further cycles of hair growth. making over time can bring measurable benefits to many the people. Researchers explain that the bellows they create will make them better study of drugs that stimulate hair growth and even to the regeneration of the lost the bellows. In addition, the acquired knowledge will help in researching the development of others the organs. The Japanese team is announcing another key step. In the next stage, we want to use human cells and work on drugs and methods in the field of regenerative medicine, says Professor Junji Fukuda, one of the researchers. Scientists have printed a cheesecake. 3D printing technology can revolutionize the kitchen. Scientists showed its potential in this field. They printed cheesecake. How to granted, the printing of food products can provide more customization food to a person's needs, improve their safety and enable users are easier to control the nutrient content of meals. 3D printing technology is used for a lot of things. With its help, houses, prostheses, implants, doctors, are created implanted the woman with an ear printed from her own cells, and even her organs, it's like a heart. Now scientists from Columbia University and Pace University they used this technology to print the cheesecake. But the first attempts not they performed best. Over time the scientists refined the recipe and created the cake that resembles the work of a cook. Whether ovens, pans, self-various, or microwave ovens will they go to hell? Well, maybe it is. This is likely to happen with 3D printers. However, this will not happen anytime soon. Scientists from New York they tried to print the cheesecake. We did it, but when it comes to the quality's taste, it didn't work out well. The first 3D printers to print food were quite adapted a long ago. In 2005, in the Hoda Lipson laboratory at Columbia University the first experiments were conducted. So far, this technology has been limited to creation of two-component food, which was a significant limitation. In new research, Jonathan Bluttinger's team designed special three-dimensional printing system capable of constructing meals with 7 different components, retrofitting the 3D printer in nozzle designed for squeezing selected ingredients and program the printer to DOS to them the dishes are prepared. They also armed the equipment with a laser that allows for machining the thermal components. In the case of a cheesecake, which took 30 minutes to print, scientists used cracker paste, peanut butter, strawberry jam, Nutella, banana puree, cherry sprinkles and vulnera. The results of the first attempts were not satisfactory. However, from sometimes the scientists refined the recipe and after 5 unsuccessful attempts arose a cheesecake that resembled a real cheesecake. How did he taste? Definitely it tasted like something I had never tried before. It was even good, but it's not a conventional mixture. We are not Michelin chefs. He pointed out Blutinger, the lead author of the publication. The most successful recipe used crackers as basic component of each layer of dough. Peanut butter and Nutella sumptuous the best is support layers that created pockets for more soft ingredients, banana puree and jam. In this way the cake obtained the rigid structure and did not collapse. Cheesecake is the best thing we can do now present, but our printer can do a lot more. We can print chicken, beef, vegetables, and cheese. Anything that can be turned into a paste, liquid, or powder, said Blutinger. Blutinger believes that printed food is natural consequences of the development of this technology in confrontation with the analog world stove, steamers, and pans. Potentially, such equipment can be found application almost everywhere, from food producers to restaurants and houses. 3D printers can be the next step in automation, freeing us from the need to prepare dishes. Blutinger sees this technology as a good way for calorie tracking and precise control of nutrients, helping in preparing food for people on various diets. And although the authors admit that the food from the printer should be qualified as highly processed foods that, as many have shown research, is fatal to health, it also claims that the presented by them the approach to cooking can help solve this problem because it is possible precise calibrating the nutrient and calorie content in food. We have a huge problem with low nutritional value the processed food. 3D food printing will continue to mean processed foods, but perhaps for some people a positive news it will allow for better control and adaptation of nutrition to specific. His Cooper of Pace University Nutrition and Dietetics, said Kristen Cooper of the Foods and Dietetics. However, this technology is still in its infancy. It still has a long way to go on the market. Because food printing 3D is still a nascent technology, it needs an industry ecosystem supporters, such as food cart rides manufacturers, or even authors regulations for this type of devices. Bluding pointed out. Ariel, one of the moons of Uranus, may have a subsurface ocean. Recent observations using the James Space Webb suggest that beneath the surface of Ariel, one of Uranus' moons, there is a hidden ocean. Using the James Webb Space Telescope, astronomers gathered the emission spectra of Ariel and compared them with the spectra simulated in the laboratory, the chemical mixtures. This has shown that the moon has rich deposits of dioxide, the coal. It's intriguing, because in the cold regions of the solar system where it circulates uranium, 20 times farther from the sun than the Earth, carbon dioxide easily converts in gas and escapes into space. This, in turn, means that a process must deliver CO2 to the surface of Ariel. Until now, the most popular concept has been enjoyed by the concept that the interaction between the moon's surface and the charged molecules in Uranus' magnetospheres create carbon dioxide through a process called radiolysis. However, a new study published in the astrophysical journal Letters indicate that carbon dioxide and other particles get out of inside the moon, perhaps from a subsurface ocean. Ariel is one of the 5 largest moons in Uranus. Although 28 natural satellites are circulating around the icy giant, at least about that's all we know. The 5 largest are named Titania, diameter approximate 1,578 kilometers, Oberon, diameter approximate 1,522 kilometers, Umbriel, diameter approximate 1,169 kilometers, Ariel, diameter approximate 1,158 kilometers, and Miranda, diameter approximate 471 kilometers. Ariel was discovered with Umbriel in 1851. By William Lassell. Its name was taken from 2 different literary works, from the play of William Shakespeare The Storm and poem Pupil Kidnapped by Alexander Pope. In 1986, he flew in the vicinity of Uranus. Voyager 2 took pictures of Ariel, and these are so far the only high-resolution photographs of the moon we have. These images showed the crater-covered surface of Ariel and deep and long the ravine. Researchers believe Ariel is made up of ice and materials, the rocks. Its surface is relatively young and shows signs of activity geologically driven, most likely by tidal heating. In the studies published last year have been decided that on the moon it could to be water. Uranus in the solar system is distinguished by its rotation axis, which it's very inclined. Its poles lie where most of the planets have an equator. Ariel circles Uranus in the equatorial plane, which is almost perpendicular to the planet's orbits. Chemical elements and molecules absorb and emit light with characteristic wavelengths, leaving individual fingerprints on emission spectra. With the Webb telescope, the team research by Richard Cartwright of Johns Hopkins Applied Physics Laboratory collected the moon's spectra, which allowed to determine its composition in detail. The chemical comparison with simulated spectra from chemical mixtures prepared in a laboratory on Earth revealed that Ariel has one of the richest carbon deposits in the solar system. What's more, in the spectra of the researchers found chemical signatures of carbon monoxide. He shouldn't be there. You have to go down to 30 kelvins, minus 233 degrees Celsius before carbon monoxide becomes stable, and Cartwright said. Meanwhile, the average surface temperature of Ariel is higher by about 18 degrees Celsius. Carbon monoxide must be actively replenished, no doubt, the researcher added. Cartwright admitted that radiolysis could be responsible for some. This addition is. Laboratory experiments have shown that bombing radiation water ice mixed with carbon-rich material can produce both carbon dioxide and carbon monoxide. However, the observations from Voyager 2 flyby and other recent findings suggest interactions. The standing behind the radiolysis may be limited because Uranus's magnetic field axis and the plane of the orbit of its moons are shifted relative to each other by about 58 degrees. Scientists believe that most of the carbon monoxides may be from chemical processes taking place in the subsurface ocean. The compounds of these they get to the surface of Ariel probably through cracks in the ice shell of the moon. Moreover, new observations suggest that the surface of Ariel it also contains carbonate minerals that can only be created through interaction of liquid water with rocks. prevalence of heart attacks and strokes lower after vaccination against COVID-19. Research involving 46 million adults found that after vaccination against COVID-19 has decreased the incidence of heart attacks and strokes. The new analysis covered almost the entire adult population of England. In the research by researchers from the universities of Cambridge, Bristol, and Edinburgh, supported British Heart Foundation checked medical records 46 million the British from December 8, 2020 to January 23, 2022. Scientists compared the incidence of cardiovascular disease before vaccination against COVID-19 and 2 years after the introduction of the vaccination program. Results published in Nature Communications showed that the incidence of heart attacks and strokes was up to 10% lower in 13 to 24 weeks after the first dose of the COVID-19 vaccine. After the second dose, prevalence was up to 27% lower after receiving the vaccine AstraZeneca and up to 20% lower after the Pfizer-BioNTech vaccine. The frequency, the occurrence of other clots, mainly pulmonary embolism and venous thrombosis deep lower limbs, showed a similar pattern. We have tested COVID-19 vaccines and disease cardiovascular in nearly 46 million adults in England and found a similar or lower incidence of common diseases cardiovascular, such as heart attacks and strokes, after each vaccination than before or without vaccination. These studies additionally confirm large number of evidence for the safety of the COVID-19 vaccination program, which, as has been shown, provides protection against the severe course of COVID-19, and he saved millions of lives around the world, said Dr. Samantha IP from the University of Cambridge, co-author of the study. As we read in the message published on the pages the University of Cambridge, previous studies have shown that the prevalence rare cardiovascular complications are higher after some vaccines against COVID-19. Myocarditis has been reported and pericarditis after mRNA-based vaccines such as vaccine Pfizer-BioNTech. The incidence of thrombocytopenia induced has also been reported AstraZeneca vaccine. The new analyses confirm these findings, but what important, no new cardiovascular diseases identified in them vaccination related to COVID-19, which indicates that the benefits vaccines outweigh the risks. The prevalence of cardiovascular diseases is higher after COVID-19. This is especially true for severe cases. Maybe this explains why the incidence of heart attacks and strokes is lower in vaccinated people compared to unvaccinated, but more the explanations go beyond the scope of this study. COVID-19 vaccination program has begun in UK with momentum, and more than 90% of the population over 12 was vaccinated at least one dose by January 2022. This is a test covering all of England to provide safety assurance to patients cardiovascular first, second, and dose-like vaccine against COVID-19. It shows that the benefits of the second and dose-like, with fewer common cardiovascular events, such as heart attacks and strokes after vaccination, exceed very rare complications. Cardiovascular Professor William Whiteley of the University of Edinburgh. Given the critical role of vaccines in protection people before COVID-19, it is important that we continue to explore the benefits and risks of they are tied up. The availability of data covering the entire population allowed us examine various combinations of COVID-19 vaccines and consider rare the cardiovascular complications. This would not be possible without very large data, to whom we have the privilege of access in our close cooperation interinstitutional, said Dr. Vanexia Walker of the University of Bristol. The origin of the flores. Homo floresiensis is the smallest of our ancient relatives. Scientists estimate that it has reached only a meter tall. New uncovered fossils, a bone of 700,000 years ago, including the smallest arm bone that ever they found, suggest that a species colloquially referred to as a hobbit from Flores, it evolved from our importance of greater relatives. In 2003, in one of the caves on its own to Indonesia Flores Island, remains of extinct hominida species have been discovered. It is called Homo floresiensis. Thus the smallest of our relatives. Scientists believe it was only about a meter high. Because of this species is also referred to as a hobbit from Flores. Discovery of bone in Liang Bua cave on Flores surprised the scientific environment. Researchers have long wondered whether they were dealing with the remains of the previously unknown hominida or grubbed-up species, a representative of another of our relatives. In the end, it turned out that the smallest of our ancient ancestors. almost complete skeleton found in the Liang Bua cave women and the remains of 8 other representatives of this species. The date of they indicated that they are about 50,000 years old. It is known that at this time Homo sapiens were already settled in Australia and Homo erectus lived a few hundred kilometers away on the island of Java. Perhaps it is representatives of these species reached the island and met small hobbits. It is possible that they were responsible for their extinction. In 2014, teeth were discovered in another part of the island, a fragment jaws and stone tools from 700,000 years ago, which also seem to it belongs to Homo floresiensis. However, these parts of the body are not the best to determine the growth of the individual to whom they belonged. But recently scientists have found 3 new hobbit fossils from the same period. Among these are the smallest arm bone ever found. Although for example partial, is very important and sheds new light on the origin of Homo floresiensis. In the research published in the journal Nature Communications, scientists have shown that the hobbits probably came from Homo erectus and its faint size evolved relatively shortly after arriving on Flores Island. Studies have shown that tiny arrogant bone found on Mammoth's position is from an adult. Mammoth is located around 75 kilometers from the cave of Liang Bua. It's a sparsely grassy island. It was there in 2014 that the teeth and fragments of the Homo floresiensis jaw were discovered. They belonged to at least 3 individuals with jaws and teeth a bit smaller than Homo floresiensis, suggesting that small size the bodies evolved early in the history of Flores hominids. In the course of research some of the teeth found were considered to be an intermediate between early Homo erectus and Homo floresiensis, but because of a small number the specimens were not entirely clear to which species they belonged. Newly discovered bones can solve the mystery of origin hobbits from Flores, especially the lower half of the framed bone. It belonged to an adult. Based on the estimated bone length, the team was able to calculate the body height of this hobbit at about 100 centimeters. That's about 6 centimeters less than estimated height of the skeleton body from the cave of Liang Bua. This rime of an adult individual from over 700,000 years it is not only shorter than Homo floresiensis, it is the smallest bone-a-rimed hominid known from the fossil record, said Professor Adam Brum from Australian Research Centre for Human Evolution at Griffith University, co-author of the publication. This very rare specimen confirms our hypothesis that the ancestors of Homo floresiensis were extremely small in terms the size of the body. The small proportions of this limb bone clearly show that the early ancestors of the hobbits were even smaller than we thought, he added. Bones from Mammoth's office make up a total of 10 specimens fossil and come from at least 4 individuals, including 2 children. They are all very anatomically similar to Homo floresiensis with Liang Bua and can be considered an older variety of this species. However, although these specimens belong to the ancestral ancestors of the Liang Bua, this earlier form had less a specialized period. In addition, scientists argue that from the tiny bone the shoulder appears that extreme body size reduction occurred early stage of the history of the Flores. The evolutionary history of the Flores hobbits is still in the big meals unknown, said Professor Brum. However, new fossils they strongly suggest that the history of the hobbit actually began when the group early Asian representatives of Homo erectus in some way was isolated on this remote Indonesian island, perhaps a million years ago, and over time, she went through a drastic reduction in body size, he added. Fossils from Mammoth showed that it is extremely small body size Homo floresiensis has evolved in the first 300,000 years of their history on the island, and then lasted for over 600,000 of years. Why did that happen? It's a tough question, said Yusuf Kaifu from Toki University, co-author of the study. Researchers point to the phenomenon of island dwarfism, in which species reduce their size as they live in isolated locations. This is a well-known evolutionary trend, probably due to limited resources on the island. Large body sizes are needed to fight predators, but when this factor does not occur, it seems that evolution promotes the smaller the size. In Liang Bua, scientists also found Stegolin's remains, extinct a relative of elephants with an increase of only 1.5 meters, or 30% of its size on the continent. Ancient viruses hidden in DNA may promote development the cancers. Endogenous retroviruses are viruses that thousands of years ago they infected people, The trace of these infections is visible in our genome to today. For a long time, it was believed that the genetic material of these retroviruses is junk DNA, but in new research, scientists showed that when retroviruses endogenous will be reawakened, can play a key role in development the cancers. Research also suggests that silencing some of them may be quite improve the effectiveness of the disease treatment. It is believed that about 8% of the modern human genome it consists of a sequence of endogenous retroviruses, HERV, human endogenous retrovirus, which thousands of years ago infected our ancestors. Theirs genetic material was passed on to subsequent generations and permanently incorporated to the genetic material of man. For a long time, scientists thought it was ancient viral DNA is inert and does not have any significant function in the body. It is not. He is also able to cause damage. However, in new research, researchers from the University of Colorado in Boulder, they showed endogenous retroviruses contribute to cancer development and their silencing may increase the effectiveness of therapy the anti-cancer. Results and description of the research appeared in the journal Science Advances. Among the approximately 20,000 genes that serve as the building blocks of life, there are also DNA molecules left by viruses that they infected our ancestors in ancient times. Our study shows that today's diseases can largely be due to the ancient ones' virus infections, which until recently, few researchers have pointed out, Edward Chuang of the University of Colorado at Boulder said. Studies show that about 8% of the human genome is depositing from endogenous retroviruses that slip into our cells, the evolutionary ancestors. In time they penetrated into the sperm, eggs and embryos, which resulted in their genetic material being transmitted to the next generation. In May this year, a work was published showing that some of these retroviruses increase susceptibility to mental disorders such as schizophrenia, bipolar disorder, and depressive disorder. The new studies have shown that endogenous retroviruses can act as switches, which includes nearby genes. Some of them may contribute to our immune response to modern viruses such as COVID. Chuang and Atma Ivancevic investigated the role of retroviruses endogenous in cancer. They analyzed published sets data on epigenomes of 21 different types of cancer. They found out that it is concrete the line of endogenous retrovirus known as LTR10, which infected the ancestors the chiefs about 30 million years ago, showed surprisingly high levels activity in several types of cancer, including lung and colon cancers. Further analysis of dozens of patients with colorectal cancer showed that LTR10 was active in about a third of them. When researchers used a CRISPR gene editing tool to cut or mute the sequences in which the retrovirus was present, found that key genes that are known to promote cancer development, also, they're gone. Experiments on mice have produced similar results. When the researchers removed the LTR10 switch from cancer cells, key promoting genes the development of the disease, including one called XRCC4, has also been excluded, and the treatment was better effective. We know that cancer cells express many genes that they should be on, but no one really knows what it is turning on. It turns out, many of the switches come from ancient viruses, Chuang said. The authors of the publication note that only this one family retroviruses are regulated by 70 genes associated with cancer in the so-called MAP kinase trail, cellular path that is unfavorable reprogrammed in many cancers. Different families of endogenous retroviruses, they probably affect the different pathways that promote different cancers. Chuang said that although these viruses are probably an underestimated source of genetic impact on all types of cancer, the test does not show that they cause cancer. According to him, the cancer awakens them, giving them the opportunity to activate the processes conducive diseases to help keep him alive. The researcher also suspects that with aging human cells are formed, their defense mechanisms weaken, allowing the ancient viruses to reawaken and contribute to other problems' health. The origin of diseases in the cell has always been a mystery. Endogenous and endogenous retroviruses are not the whole story, but they can make up a big part of it, he said. Chuang. The first successful titanium heart transplant. The 58-year-old from the United States became the first person in the world to be the ill and inefficient heart was replaced with a titanium heart. The device using magnetic levitation technology, maglev, the same one that is used in high-speed trains. The Titanic Authority is temporary solution and to give more time waiting for real transplant the heart. The titanium heart was developed by VACOR. Representatives of companies say their heart is strong enough to keep a man with life even during exercise. In addition, its size is small enough for the titanium organ to fit most men and women. The two-chamber device has a fist size and is it's indestructible. It is resistant to corrosion and mechanical wear. The Titanic heart, it doesn't beat, but it can pump blood for many years. With its design, scientists have abandoned almost all moving parts. All this to make them less unreliable. Device using magnetic levitation technology, the same one that is used in high-speed trains. His only movable part is magnetically suspended double-sided rotors with left and right waistbands placed in two separate pump chambers. The technology of maglev, it allows pulsed blood pressing through fast rotor cycles. The device can pump blood in the so-called small and large circulation. In the large circulation pumps heavily oxygenated blood from the heart to the rest of the body. We say here's about oxygenated blood. In a small circulation of pistons, deoxidized blood returning from the whole body to the lungs, where gas exchange takes place, blood gives away the dioxide carbon, and it takes oxygen. The whole device is powered by a small, external, the portable controller. Work on the titanium organ took many years, but artificial the heart finally went to the chest of a live patient with insufficiency the heart in the final stage. The device was implanted in Baylor St. Luke's of the Light Medical Center at the Texas Heart Institute. The surgery went without complications. The patient, 3 days after the titanium implantation of the organ, freed from the ventilator. On the 7th day after the operation, he could walk 150 meters. In total, the man lived with the titanium heart for 8 days. During this time, the donor was and the artificial organ has been replaced by a true heart. I'm incredibly proud to witness the first implantation of our artificial heart in man, said the founder by VACOR, Daniel Timms. This achievement would not be possible without our courage first patient and his family, dedication to our team and ours experts working with the Texas Heart Institute, he added. Currently, the best treatment option for people with severe heart failure is a transplant, but not always the heart is available and it's in good time. Artificial hearts are a way to extend and improve quality life of patients eligible for transplantation who are at risk of dying and waiting for a donor. No one really knows how long the titanium device it can function in people. In the VACOR laboratory similar to the implanted, the artificial organ has not stopped spinning for 4 years. The company has obtained approval from the FDA to implant its body 5 patients with life-threatening heart failure who have been qualified for transplantation and are waiting for the organ. Heart failure affects at least 26 million people worldwide. Every year he performs globally less than 6,000 heart transplants. Subsequent remains of insects and plants were found under Greenland ice. Scientists analyzing sediments excavated from the Greenland sheet, from a depth of more than 3 kilometers, they found the remains of mushrooms, insects and the plants. Research provides evidence that in the recent geological past does not only the edges, but also the central part of the island ice sheet melted. If it had happened this is again, according to estimates, the level of the seas and oceans would increase by 7 Mammoth. Research published in the journal Proceedings of the National Academy of Sciences focused on the GISP2 ice core. Extracted in 1993 from central Greenland. At least before this material was studied, it was then that scientists did not look for fossils and settlement from under the ice. New analysis of the core drilled 30 years ago resulted in discovery ancient plants, seeds, mushrooms and insects preserved under the Greenland sheet. Research shows an island over the last million years was an Arctic free ice tundra full of insects and plants. This was at a time when the level of carbon in the atmosphere was much lower than it is today. These findings indicate that the Greenland ice sheet is more vulnerable to climate change, which has been previously thought and has potential for significant raising sea levels and oceans. In GISP2 core sediments, scientists found, among others, soil containing willow wood, residues of various insects, mushrooms or seeds in excellent condition. Research confirms Greenland ice it melted and the island greened during the warm period that took place probably over the last million years. This suggests that the island's giant ice sheet is more fragile than scientists thought. "We saw the fossils in the first hour, maybe half an hour, work on them," said Paul Bierman of the University of Vermont, one of the authors of the publications. Researchers found in a several centimeter layer soil willow wood, plant remains with for the four cuters, the remains of mushrooms, the eye of the complex insect and the poppy arctic, only one seed. "These fossils are beautiful," Bierman added, indicating at the same time what this means with respect to the impact caused by human climate change to melt the Greenland sheet. If the ice covering the center of the island has melted, it shall be asked to assume the rest of the ice sheet split that fate. Scientists indicate that this condition probably lasted for many thousands of years. It's enough time for different ecosystems to form. This is what the study confirms, that a significant increase in sea level occurred in the time when the causes of warming were not particularly extreme, which is warning of damage we can cause if we still warm the climate, said Richard Alley of Pennsylvania State University, who reviewed the new research. Currently, sea level rises by about 2.5 centimeters per decade, but as Bierman pointed out, warming accelerates, and with it growth sea level and oceans. The scholar added that if greenhouse gas emissions are not, it will be radically reduced, almost complete melting of Greenland over the next centuries. It will lead to sea level rise by about 7 Mammoth. Bierman warned not to buy a house near the coast. Look at Boston, New York, Miami, Mumbai, or choose your own a seaside city in the world and add more than 7 meters of sea level, he said. Bierman. In 2016, Jörg Schaefer from Columbia University he studied the GISP2 core by focusing on the rocks. They have published controversial analysis at the time suggesting that the current ice sheet of Greenland must not be more than 1.1 million years old, and that in the Pleistocene period, geological, which began 2.7 million years ago, on the island occurred longer periods without ice. Scientists noticed then that if the ice melted at the core intake site GISP2, would also be melted at 90% of the rest of Greenland. It was an important step in the direction of refusing the long-standing belief that Greenland is frozen from its millions of years. In 2017, Bierman, co-author of the study, he and an international team of scholars analyzed the ice core excavated in near the coast of Greenland in the 1960s. The 20th century. In this research he discovered twigs, seeds, and parts of insects, revealing that ice in this place has melted in the last few 100,000 years. Although the ice in rocks from the core has been thoroughly studied, no one looked at the several centimeter layer of clay at the end of the core to check if it is soil and whether it contains plant remains or the insects. This prompted the Bierman to reach for the core of the GISP2, which was stored at the National Science Foundation Ice Core Facility in Lakewood, Colorado. New GISP2 core research seems to confirm that the hypothesis in 2016 is correct. Moreover, they show that the island was warm enough, for a long enough time for vegetation, perhaps even a forest with dwarf trees, developed where the ice today is over 3 kilometers deep. We now have direct evidence that not only ice on Greenland disappeared, but that there were plants there and the insects. This is indisputable, Bierman said. If the ice in the center of the island decided, almost certainly not at the rest of Greenland, which means trouble for the world today powered by fossil fuels. Hundreds millions of people around the world will lose their place to life, he warned the researcher.